Welcome to the Late Lab. It's lovely to have you here. Before settling in, where are you listening from tonight? Are you stretched out in bed at the end of a long day, curled up on the sofa with a blanket and something warm to drink, travelling home in the dark with the world outside reduced to passing lights and reflections? Wherever this finds you, the hope is that you're safe, comfortable, and ready to drift gently into thought. If you enjoy these slow and sleepy journeys through science, a like and subscribe really helps this little channel keep drifting along. And if you've already subscribed and come back regularly to spend these quiet evenings here, thank you. Your support means a great deal, and it genuinely helps keep this calm little corner going. Now, there is always the possibility that tonight's subject might be just interesting enough to keep sleep waiting a little longer. But if that happens, at least there will be the small reward of spending those extra waking minutes thinking about hidden oceans, distant atmospheres, and the strange challenge of working out whether a faraway world is merely habitable or truly alive, which, all things considered, is not the worst reason to be awake. So let's begin, shall we? How would scientists actually know if there's life beyond Earth? To search for life beyond Earth, the first problem is almost surprisingly basic. Before anyone can hope to detect alien life, there has to be some clear idea of what life actually is. That sounds like the sort of question science should have settled neatly a long time ago. But the closer it is examined, the stranger it becomes. Life seems obvious when looking at a bird, a tree, or a fish. It becomes much less obvious when looking at a virus, a spore, or a crystal that grows in a cave with no help at all from biology. That is because life is not defined by one single magical property. It is usually understood as a cluster of traits that tend to appear together. Living things use energy. They maintain internal order, they store information, they respond to their surroundings, they reproduce, or belong to systems that reproduce. They evolve over time through natural selection. One trait on its own is not enough. A fire spreads and consumes energy, but it does not carry inherited instructions the way a cell does. A crystal can grow, but it does not adapt through evolution. A virus contains genetic material, but outside a host cell it is strangely inactive, caught in an awkward grey zone between chemistry and biology. Modern biology therefore treats life less as a simple label and more as a process. A living thing is not just a collection of molecules, it is a system that takes matter and energy from its environment and uses them to keep itself organised. That is important because the universe generally leans toward disorder. Molecules break apart, gradients fade, and structures decay unless energy keeps them going. Life resists that drift. It builds and repairs itself. It preserves an internal pattern. In a sense, life is matter that keeps insisting on staying interesting. One widely used working definition from NASA describes life as a self-sustaining chemical system capable of Darwinian evolution. It is not a perfect definition, but it is useful. It includes chemistry, which matters because life must be built from real materials rather than mystical force. It includes self-sustaining behaviour, which helps separate living systems from things that only behave biologically when something else does all the work. And it includes Darwinian evolution, which points to one of the deepest features of life. Once it exists, it changes over generations through variation and natural selection. That idea reaches back to Charles Darwin, whose work transformed biology. Darwin did not explain how life first began, but he did show how living populations change once they exist. Organisms vary. Some variations help survival or reproduction. Those traits become more common over time. Across enough generations, life adapts to its environment. This matters for astrobiology because the search for life is not mainly a search for familiar creatures. It is a search for organised systems that use energy, preserve information, and change through evolutionary history. Metabolism is one of the clearest features of that process. Living things do not merely sit there looking decorative. They take in energy and use it to power chemical reactions. Plants capture sunlight. Animals consume other organisms. Microbes, as usual, are more inventive than everyone else. Some microbes use oxygen, others avoid it. Some gain energy from sulfur, iron, hydrogen, or methane. Around hydrothermal vents on the ocean floor, entire ecosystems survive without sunlight, relying instead on chemical energy released where hot rock meets seawater. That alone shows that life does not have to resemble a sunny meadow to be real. Information storage is just as important. On Earth, living cells carry instructions in molecules such as DNA and RNA. Those molecules preserve the patterns needed to build proteins, control chemistry, and pass traits from one generation to the next. This means life is not just active chemistry, it is chemistry with memory. It can preserve a working design, modify it, and hand it onward. That is one reason a bacterium is alive while a storm is not. A storm can be dynamic and complex, but it does not store a coded plan for producing more storms with inherited variation. Reproduction matters as well, though even here nature refuses to be perfectly tidy. A mule is alive despite usually being sterile. Worker ants may never reproduce, yet they belong to living colonies. Seeds and spores can remain dormant for astonishingly long periods and still return to full biological activity. So it is often better to think not only about individual reproduction, but about membership in a lineage. Life belongs to systems that, in some form, can produce continuity over time through inherited information. All of this begins to show why the title question is harder than it first sounds. If scientists want to know whether something on another world is alive, they need criteria that are broad enough to include unfamiliar biology, but strict enough to avoid mistaking non-living chemistry for life. That is not easy, because the universe is already capable of astonishing chemical complexity without any biology at all. Organic molecules exist in interstellar clouds. Comets contain carbon compounds. Titan has lakes of methane and a rich atmospheric chemistry. None of that is trivial, but none of it is proof of life either. Chemistry can do a great deal on its own before biology enters the story. This is why astrobiology is such a careful science. Astrobiology brings together biology, chemistry, geology, astronomy, and planetary science to ask not just where life might exist, but how it could be recognized honestly. Biology helps define what life does. Chemistry shows what matter can build without life. Geology reveals what rocks and planets can preserve. Astronomy identifies potentially habitable worlds. Planetary science asks how those worlds change through time. All of these fields are needed because detecting life is not the same as detecting water, carbon, or a promising temperature. That distinction matters enormously. Detecting a living organism directly is one thing. Detecting the environmental effects of life is another. Detecting conditions that might allow life is something else again. These should not be confused. Finding liquid water on a distant moon would be exciting, but water is not life. Finding organic molecules on Mars would be important, but organic molecules are not life. Even finding oxygen in the atmosphere of an exoplanet would not automatically prove life, because under some conditions oxygen can be produced through non-biological processes. A habitable world is not necessarily an inhabited world. Earth itself offers a useful warning. For a large part of its history, life existed here long before the atmosphere became rich in oxygen. For billions of years, this planet may not have looked dramatically alive to a distant observer, even though microbes were already thriving in oceans and sediments. A living world does not always advertise itself clearly. It may be biologically active but visually quiet. It may hide its life underground, beneath ice, or under an ocean surface. It may produce only faint signals, easy to miss and easy to misread. So when scientists ask what counts as life, they are not asking a philosophical question for amusement. They are building the rules for one of the most difficult detection problems in science. They need a way to distinguish organized, evolving, energy-using systems from everything else that planets and chemistry can produce. They need to know what signs would point toward real biology, what signs might be misleading, and what kinds of life might remain almost invisible. Life, in the scientific sense, is matter doing something unusually persistent and structured. It uses energy to maintain itself. It stores information. It produces continuity. It changes through evolution. It creates patterns that chemistry alone does not easily sustain. That does not make the definition simple, but it does make the search possible. And if something like that exists elsewhere in the universe, then recognizing it will begin here, with the difficult task of deciding what life is before trying to find it among the stars. Every serious search for life beyond Earth begins with a slightly humbling fact. However wide the universe may be, however many planets circle distant stars, however many icy moons hide dark oceans beneath frozen crusts, there is only one world where life is known to exist with certainty. That world is Earth. It is the only confirmed example, the only place where chemistry unquestionably crossed the line into biology, and for that reason, it quietly sits behind almost every idea scientists have about alien life. When researchers imagine what life elsewhere might need, what traces it might leave, or how it might survive, they are almost always starting with the one successful case already available. This does not mean Earth is assumed to be ordinary. In truth, no one yet knows how ordinary or extraordinary it may be. It simply means that Earth is the only planet where the full chain is visible. Here, matter organized itself into cells, cells diversified, evolution unfolded, and biology eventually spread into oceans, rocks, air, and soil, with the confidence of a process that had clearly found a durable foothold. If there are other living worlds, they may be quite different. But until one is found, Earth remains the reference point, the example in the margin, the only known proof that a living planet is possible. That makes early Earth especially important. Not Earth as it is now, with forests, oceans, insects, whales, and an atmosphere full of oxygen, but Earth in its much younger and stranger state. The early planet was not a calm blue refuge waiting politely for life to appear. It was a restless world shaped by impacts, volcanism, shifting crust, intense weather, and oceans that may have formed surprisingly early. Liquid water seems to have been present very far back in Earth's history, and that matters because water is an unusually effective medium for chemistry. It allows molecules to move, collide, dissolve, react, and reorganize. It is not a magical life potion, but it is chemically useful in almost rude abundance. Alongside water, early Earth offered energy. This is crucial. Life cannot emerge or persist without access to energy gradients, places where one chemical state can fall into another and release useful power along the way. Sunlight provided one source, volcanic systems provided others. Lightning may have contributed some local chemistry. Deep beneath the surface, water interacting with hot rock could create chemical disequilibria rich in potential. Hydrothermal environments, whether shallow volcanic pools or deep ocean vent systems, remain among the most discussed candidates for life's early setting, because they combine water, minerals, heat, and chemical gradients in one place. In simpler terms, they offer ingredients, surfaces, and a constant supply of opportunities for chemistry to become more elaborate. Mineral surfaces may also have mattered more than they first sound. Clay minerals, metal sulfides, and other crystalline materials can help concentrate molecules and encourage reactions that would be less likely in open water alone. In origin of life research, this is one reason bare rock is treated with more respect than it usually receives. A mineral surface can act a little like a workbench, holding molecules in place long enough for something more interesting to happen. That does not mean a rock is halfway to becoming a cell, which would be an alarming precedent. It means the physical environment may have helped guide chemistry toward greater complexity. Even with these promising conditions, the exact pathway from non-living chemistry to living systems remains unresolved. This is one of the most important honest limits in the field. Scientists do not yet know precisely how life began on Earth. There is no complete step-by-step record, no preserved sequence showing the moment when a loose collection of molecules became a self-sustaining, evolving system. There are hypotheses, experiments, promising routes, and plausible environments, but no final answer. That uncertainty matters because it means any search for life elsewhere must proceed without pretending that the origin of life is already fully solved. The general term for life arising from non-living chemistry is abiogenesis. It does not refer to spontaneous generation in the old mythical sense, as though mice simply appear from piles of laundry when left unattended long enough. It refers to a long chemical transition in which simple compounds become more complex, more organized, and eventually capable of replication and evolution. At some point in that transition, chemistry becomes biology. Exactly where that boundary lies is still debated, but the broad concept is clear enough. Life emerged from matter that was once not alive, and Earth is the only place where that has definitely happened. One influential line of thought is the RNA world hypothesis. This proposes that before modern cells used the full biological partnership of DNA, RNA, and proteins, there may have been an earlier stage in which RNA played a double role. It could store information and also help catalyze chemical reactions. That is attractive because one of the hardest problems in origin of life research is explaining how heredity and function became linked. Modern biology separates those tasks across different molecules, but early life may have relied on something simpler. The RNA world idea does not solve everything, and it may describe only one stage rather than the entire story, but it remains central because it offers a plausible bridge between chemistry and evolution. The work of Stanley Miller and Harold Urey became famous for related reasons. In the early 1950s, their experiments showed that under certain conditions, simple chemicals could produce amino acids, some of the building blocks used by life. The exact atmospheric assumptions in those original experiments are no longer taken as a perfect model of early Earth, but the broader lesson survived. Prebiotic chemistry can generate biologically relevant molecules without any organism being present. Since then, origin of life research has expanded far beyond those early flasks, with laboratory studies exploring lipids, nucleotides, catalytic surfaces, hydrothermal systems, and networks of interacting reactions. Institutions linked to astrobiology and prebiotic chemistry, including NASA-supported research groups and major university laboratories, continue to test which steps are chemically plausible and which are not. Once life did begin, another distinction becomes important. The origin of life and the later evolution of life are not the same problem. Abiogenesis asks how the first living systems appeared. Evolution by natural selection explains how those systems diversified afterward. By the time there was a last universal common ancestor, often shortened to LUCA, life was already well underway. LUCA was not the first life form, but the most recent shared ancestor from which all known life on Earth descends. That means everything alive today, from oak trees to octopuses to bacteria living in boiling springs, traces back through a common biological history. The existence of LUCA tells us that life on Earth, for all its variety, is a single family. That family resemblance shapes every search strategy used elsewhere. Because Earth is the only confirmed living world, scientists begin by asking what Earth life does to a planet. It changes atmospheres. It alters rocks. It leaves organic molecules, isotopic patterns, microfossils, pigments, gases and textures. It creates signatures in oceans, sediments and air. Some of those traces can be preserved in stone. Some might be visible through a telescope. Some might drift from an icy moon in a frozen plume. The reason researchers study Earth so carefully is not because they assume alien life must copy it exactly, but because Earth provides the only tested example of how living chemistry can announce itself to the wider world. So before scientists search distant planets, icy moons, or buried Martian sediments, they return to this one known case. Earth is not merely home. It is the control sample, the confirmed success, the only world where the full story from chemistry to biology is known to have happened. Everything else begins there. If life exists beyond Earth, it may not introduce itself politely. It may not wave, glow, or arrange stones into helpful messages. Far more likely, it would leave behind traces, a gas in the air that should not be there in that amount, a pattern in rock that seems too organized, a set of molecules that suggest chemistry has been nudged again and again in a direction that ordinary geology struggles to explain. These traces are called biosignatures, and they sit at the heart of one of the most careful scientific searches ever attempted. A biosignature is any measurable feature that may be produced by life. That definition is intentionally cautious. It does not say proof. It does not say certainty. It says may. That small word matters because scientists are not looking for a magic stamp that can be read from a great distance with perfect confidence. They are looking for clues. A biosignature is a clue that biological activity might be present or might once have been present. It becomes more interesting when it appears in the right environment, in the right combination, and with fewer and fewer convincing non-living explanations. These clues can take many forms. Some biosignatures are atmospheric gases. On Earth, oxygen is the most famous example, because large amounts of oxygen in the atmosphere are tied to photosynthetic life. Methane is another, especially because on Earth much of it is produced biologically, though certainly not all. Some biosignatures are chemical rather than gaseous, including complex organic molecules or unusual distributions of certain compounds. Others are isotopic, meaning they involve the relative abundance of different versions of the same element. Life tends to process matter selectively, and that can leave subtle fingerprints in carbon, sulfur, or other elements. Then there are visual or structural biosignatures, such as textures in ancient rock, layered formations like stromatolites, pigments that alter how a surface reflects light, or seasonal changes that suggest active cycles rather than static chemistry. That range is important because life does not affect a planet in just one way. Living systems change atmospheres, reshape sediments, alter oceans, build minerals, and modify the flow of matter and energy through an environment. A planet with life is not merely a planet with organisms sitting quietly on it. Over time, life can become a geological force. Earth is the great example of this. The oxygen-rich atmosphere did not begin as a default feature. It was transformed over time by photosynthetic organisms, especially ancient microbes. Methane has flowed through the atmosphere in changing cycles. Biological activity has influenced the carbon cycle, the sulfur cycle, soils, sediments, and the chemistry of the oceans. Even from a distance, a living planet may eventually begin to look chemically unusual, but this is where caution becomes absolutely essential. A biosignature is not proof by itself. That may sound frustrating, but it is simply how good science works. Oxygen can be produced by life, but under certain planetary conditions it can also accumulate through non-biological processes, such as the breakdown of water molecules by sunlight, followed by hydrogen escaping into space. Methane can be produced by microbes, but also by water interacting with certain rocks. Organic molecules can be made by living organisms, but they can also form in space, on comets, or through ordinary planetary chemistry. A layered rock texture may hint at microbial influence, but non-living processes can also build patterns that look more persuasive than they deserve. So a biosignature only becomes meaningful in context. Context means knowing the environment in which the signal appears. What is the temperature? What kind of star does the planet orbit? What other gases are present? Is there liquid water? Is volcanic activity likely? Could photochemistry explain the signal? Is the pattern isolated, or does it fit into a wider web of evidence? Scientists do not ask only, could life make this? They also ask, what else could make this? Then they keep asking until the easy explanations have been exhausted and the uncomfortable ones have had a proper turn as well. That is why false positives and false negatives are so important. A false positive is a signal that looks biological, but is actually produced by non-living processes. A false negative is the opposite. Life is present, but the evidence is weak, hidden, or misleadingly absent. Both are dangerous in different ways. A false positive could lead scientists to announce life where none exists, which would be spectacularly embarrassing and scientifically damaging. A false negative could cause a genuinely living world to be overlooked simply because its biology does not shout loudly enough or behaves in an unfamiliar way. Earth again provides useful warnings on both sides. Today, this planet has a thick oxygen atmosphere, strong biological cycles, widespread pigments, and a fossil record. From far away, some of those signs might look promising, but Earth did not always look like this. For much of its early history, life existed here without producing an oxygen-rich atmosphere. Microbes were alive, evolving, and changing the planet long before Earth would have advertised itself clearly to a distant astronomer. That means a living world can be easy to miss. It may be biologically active and still appear quiet from a great distance. In other words, life does not always perform for the telescope. The modern science of biosignatures is built around exactly these difficulties. Research groups supported by NASA Astrobiology, the European Space Agency, and major universities spend a great deal of time modelling what living and non-living planets might look like under different conditions. Scientists such as Sarah Seeger and Victoria Meadows have done important work on how atmospheres can be interpreted, how gas combinations may strengthen a case for life, and how easily a single gas can mislead if pulled away from its planetary setting. This is one reason the field often sounds more restrained than people expect. The goal is not to be unimpressed. The goal is to avoid being fooled. One especially important idea is that combinations of biosignatures are usually more persuasive than isolated ones. A single gas might be ambiguous. A whole atmosphere in chemical disequilibrium is more interesting. On Earth, oxygen and methane exist together in a way that is difficult to maintain without continuous replenishment. Left alone, they would react and diminish. Their persistent coexistence suggests that something active is constantly feeding the system. Here, that something is life. The broader principle is powerful. Planets that look chemically out of balance may deserve much closer attention than planets that merely contain a familiar ingredient. The same logic applies to rock, ice, and oceans. A complex organic molecule in isolation is intriguing. The same molecule found alongside isotopic ratios, mineral textures, and environmental conditions consistent with biology becomes more meaningful. A seasonal atmospheric shift might be interesting. A repeating seasonal shift that aligns with temperature, surface chemistry, and other measured changes becomes much more compelling. Science grows more confident when independent clues begin to point in the same direction. This is why biosignatures are best understood not as trophies to collect, but as pieces of a case. Each piece matters, but none should be forced to carry the full weight alone. Life changes worlds, yet the universe is perfectly capable of producing strange worlds without life. The challenge is learning the difference. That means taking gases seriously, but not too seriously. Taking unusual chemistry seriously, but not romantically. Taking patterns in rock seriously, while remembering that geology can occasionally behave like a mischievous forger. So when scientists search for life, they are really searching for traces that survive skepticism. A true biosignature is not just something life could make. It is something that still looks biological after chemistry, geology, climate, radiation, and chance have all been given their chance to explain it away. That is a demanding standard, but it has to be. Because if life is out there, its signatures may be faint, partial, and mixed with noise. The task is not merely to notice something unusual. The task is to notice something unusual that keeps refusing to become ordinary. Once the search for life moves from definitions and biosignatures to actual places, the map becomes much smaller than the imagination might prefer. There may be countless worlds in the universe, but the places scientists can investigate most seriously right now are much closer to home. The first targets are inside the solar system, not because they are guaranteed to contain life, but because they can be reached, observed in detail, and in some cases sampled directly. That practical difference matters enormously. A distant exoplanet may be fascinating, but it usually appears as a faint signal in light. A nearby moon or planet can be visited by orbiters, landers, rovers, drills, spectrometers, and, if engineers are feeling unusually optimistic, machines that attempt very complicated things millions or even billions of miles away. That is why proximity matters so much. In the solar system, scientists are not limited to guessing from afar. They can analyze rocks, examine ice, sniff gases, measure mineral chemistry, map underground structure, and sometimes even collect dust or plume material directly. A world that can be reached by spacecraft becomes more than a point of interest. It becomes a test site. That does not make the search easy. Spacecraft are still limited, expensive, and not especially good at improvising when something goes wrong. But it does mean that evidence can be gathered in richer ways than would be possible from light alone. The main nearby targets are Mars, Europa, Enceladus, and Titan. Each of them is scientifically plausible as a place where life might once have existed, might still exist, or at least might preserve chemistry relevant to life. They are not identical candidates. That is one of the most important things to understand. Scientists are not applying one simple checklist and waiting for a world to pass. Each target offers a different environment, a different history, and a different style of possible evidence. Mars is a rocky planet with a past that seems to include rivers, lakes, groundwater, and a more habitable ancient climate. Europa and Enceladus are ocean worlds, where the most promising environments lie beneath ice rather than under open skies. Titan is stranger still, with lakes of liquid methane and ethane on the surface, a thick nitrogen atmosphere, and chemistry that seems almost designed to force biologists into uncomfortable honesty. Mars remains the most famous target for obvious reasons. It is nearby, relatively well studied, and geologically revealing in ways that make astrobiologists pay close attention. Dry river channels, sedimentary rocks, ancient lake beds, hydrated minerals, and subsurface ice all point to a world that was once much wetter than it is now. Billions of years ago, Mars may have had environments that looked at least intermittently friendly to microbial life. Even today, some scientists consider the subsurface a possible refuge, where briny water, chemical gradients, and protection from surface radiation might still exist in limited niches. Mars is attractive because it is a world where scientists can search for fossil biosignatures, preserved organic chemistry, habitable history, and perhaps, if fortune became unreasonably generous, signs of present microbial activity. It is also attractive because it is difficult rather than fantastical. That tends to be where the most serious science lives. Europa offers a very different possibility. This moon of Jupiter is covered by an icy shell, but evidence from the Galileo mission strongly suggests that beneath that shell lies a global ocean of salty liquid water. That conclusion is supported by magnetic measurements, surface geology, tidal flexing, and the broader understanding of how Jupiter's immense gravity needs heat into its moons. Europa is compelling because life on Earth does not require sunlight everywhere. Deep ocean ecosystems around hydrothermal vents rely on chemical energy rather than photosynthesis, and that makes dark alien oceans much harder to dismiss. If Europa has liquid water, energy sources, and the right chemistry, then it becomes one of the most important places in the solar system to test whether habitability can exist far from warm planetary surfaces. Enceladus, a small moon of Saturn, may be even more inviting in one particular way. The Cassini mission revealed geyser-like plumes erupting from the South Polar region, throwing water vapour, ice grains, salts, and organic compounds into space. That is astonishingly useful. Instead of drilling through miles of ice, which would make for a very impressive engineering proposal and an even more impressive budget crisis, scientists can sample material coming from the moon itself. Cassini found evidence consistent with a subsurface ocean and likely hydrothermal activity on the seafloor. That means Enceladus may combine liquid water, organic chemistry, and usable energy in a way that sounds almost suspiciously convenient. Not proven alive, certainly, but well worth continued attention. Titan is more unsettling in a productive way. Its surface is bitterly cold, cold enough that water behaves like rock, while methane and ethane can exist as liquids. It has rivers, lakes, rain, dunes, and a dense nitrogen atmosphere rich in complex organic chemistry. Titan may not be the most likely place for life as Earth knows it, but it is valuable because it pushes scientific thinking outward. If biology elsewhere can use unfamiliar solvents or operate in radically different conditions, then Titan helps expose how Earth-centered human assumptions may be. The NASA Dragonfly mission reflects exactly that interest. Titan is not simply a place to look for life. It is a place to test how broad the concept of habitability might really be. There are also other icy worlds worth keeping in mind, even if they receive less public attention. Ganymede and Callisto may contain deep internal oceans. Triton, orbiting Neptune, has cryovolcanic features and an unusual history. Even dwarf planets and smaller icy bodies can preserve water, organics, and internal complexity for longer than older models once assumed. The solar system is not neatly divided into one living world and a collection of inert rocks. It contains gradients, buried oceans, ancient water systems, frozen chemistry sets, and places where energy and matter may still interact in biologically interesting ways. This brings in an essential distinction between habitability and inhabited status. A world can have conditions that seem suitable for life without actually hosting life. Liquid water, energy sources, carbon compounds, and stable environments are encouraging, but they do not guarantee anything. Scientists therefore often begin by searching for habitats worth testing, rather than assuming life is present. A habitable environment is one where known life could plausibly survive. An inhabited environment is one where life actually does survive. Those are very different claims. Confusing them is one of the fastest ways to make a careful science sound more certain than it is. The organisations and missions shaping this search reflect that caution. NASA, the European Space Agency, and the Jet Propulsion Laboratory have built decades of planetary science around finding environments, measuring chemistry, and gradually narrowing the question. Mars rovers such as Curiosity and Perseverance analyse sedimentary rocks and organic compounds. The European Space Agency's Jupiter Icy Moons Explorer, often called JUICE, is designed to study the large icy moons of Jupiter, especially Ganymede, while NASA's Europa Clipper focuses on Europa's ocean potential, ice shell, chemistry and habitability. These are not missions sent to wave a banner reading Life Confirmed. They are missions built to ask disciplined questions about water, energy, composition, and environmental history. That gradual approach matters because each world demands different methods. Mars invites rovers, drilling, sediment analysis, and the search for preserved organics or fossil textures. Europa requires ice-penetrating radar, gravity measurements, surface chemistry, and study of how ocean material may reach the surface. Enceladus rewards plume sampling and the analysis of ocean-derived particles. Titan calls for atmospheric chemistry, surface exploration, and broader thinking about prebiotic processes. There is no universal test that can simply be carried from one world to another. Each candidate must be met on its own terms. So when scientists ask where they would look first, the answer is not just a list of names. It is a set of nearby worlds where life detection is scientifically plausible because real evidence can be gathered. The solar system offers accessible laboratories, each with different conditions and different chances of preserving signs of biology. Some may be empty but habitable. Some may be chemically rich yet lifeless. Some may hold only ancient traces, and perhaps one of them, buried under ice, hidden in rock, or drifting out in a frozen plume, may contain the kind of evidence that finally pushes the question of alien life out of theory and into measurement. Mars has spent a very long time encouraging optimism and then punishing it. That may be one reason it remains the most famous world in the search for life. It is close, rocky, visibly shaped by ancient water, and just familiar enough to tempt the imagination into getting ahead of the evidence. But Mars is valuable precisely because it teaches restraint. If the question is how scientists would actually know whether there is life beyond Earth, Mars provides one of the clearest answers. They would know slowly, cautiously, and only after surviving a great many disappointments. Today Mars is cold, dry, and exposed to harsh surface radiation. Its atmosphere is thin, its surface pressure is low, and liquid water is unstable across most of the planet's surface. At first glance it does not look especially welcoming. But the deeper story is written into the landscape. Orbital images and rover observations have revealed dried river valleys, delta formations, lake beds, rounded pebbles, sedimentary layers, clay minerals, and sulfate deposits. These are not vague hints. They are geological signs that water once flowed, pooled, and interacted with rock over long periods. Billions of years ago, Mars was not the desert it is now. It was a planet where water moved across the surface and where habitable environments may have existed. That matters because water is not simply useful for life on Earth. It is one of the clearest starting points for chemistry becoming biologically interesting. Ancient Mars appears to have had rivers, standing bodies of water, and mineral systems capable of preserving evidence from that wetter era. Clay minerals are especially important because they form in the presence of water and can trap or preserve organic material. Sedimentary rocks matter for similar reasons. On Earth, sediments laid down in lakes, deltas, and shallow waters are often good places to search for fossils and chemical biosignatures. If microbial life ever existed on Mars, those kinds of environments are among the best places to look for traces of it now. This is why Mars remains so central to the wider question. It shows the difference between chemistry that could support life and chemistry that proves life was there. A wet ancient lake is promising, but it is not proof. Organic molecules are interesting, but they are not proof. Methane spikes in the atmosphere are intriguing, but they are not proof. Mars keeps presenting evidence that conditions may once have been habitable, while refusing, at least so far, to hand over a clear biological signature. That tension is exactly what makes it such a scientifically important case study. The modern robotic exploration of Mars has built this picture piece by piece. The Viking missions in the 1970s were among the first to test Martian soil directly for possible biological activity. Later came orbiters, landers, and rovers from NASA and the European Space Agency, each adding detail to the planet's history. Curiosity explored Gale Crater and found strong evidence that ancient Mars included long-lived lakes, organic molecules preserved in rock, and conditions once suitable for microbial life. Perseverance, operating in Jezero Crater, is exploring an ancient delta system where sediments carried by flowing water were laid down and preserved. Jezero matters because deltas are excellent natural collectors. They gather material from a wider landscape and store it in layered deposits, which means they can preserve environmental history and potentially biosignatures. Perseverance is doing more than simply driving about and looking thoughtful. It is selecting and caching rock samples for a future sample return effort. That concept is crucial because there are limits to what a rover can do on Mars. Instruments carried by spacecraft are impressive, but they are still smaller and more constrained than the full laboratories available on Earth. Returning carefully chosen Martian samples would allow researchers to test isotopes, minerals, organic compounds, textures, and possible microstructures with far greater precision. In other words, Mars may not yield a convincing answer through one rover alone. It may require a chain of evidence carried from planet to planet and studied over many years. That need for caution has been obvious for decades. One of the most famous examples is the Viking-labelled release experiment. This experiment added nutrients tagged with radioactive carbon to Martian soil and looked for gases that might suggest metabolic activity. The results were surprising. One interpretation was that something in the soil had reacted in a lifelike way. But other instruments on Viking did not detect organic molecules in a convincing manner, and many scientists concluded that reactive soil chemistry, rather than biology, explained the result. To this day, the Viking results are still discussed because they sit uncomfortably between excitement and ambiguity. They are a reminder that even direct experiments on another planet may not produce clean answers. Another famous controversy came from a meteorite known as Allen Hills 84001. This meteorite, believed to have come from Mars and later found in Antarctica, became globally famous in the 1990s when some researchers argued that certain tiny structures and chemical features inside it might be evidence of ancient Martian life. The announcement drew enormous attention, but the claims remained contested. Alternative explanations involving non-biological mineral formation and contamination were raised, and no broad scientific consensus emerged that the meteorite contained fossil life. Once again, Mars produced something tantalizing, memorable, and unresolved. Even methane on Mars has followed this pattern. Methane is especially interesting because on Earth much of it is associated with biology, although geological processes can produce it too. Some instruments and observations have reported small methane variations in the Martian atmosphere, while others have found far less than expected or none at all. This has left scientists trying to understand whether methane is being produced locally, destroyed unusually quickly, measured inconsistently, or simply appearing in ways that are still not fully understood. It is a classic Martian story. Interesting signal, uncertain source, no easy conclusion. Mars also remains interesting below the surface. Subsurface ice is widespread, and some researchers consider the underground environment one of the best possible refuges for any present Martian life. Beneath the surface, microbes would be shielded from ultraviolet radiation, temperature extremes, and the highly oxidizing surface chemistry that makes preservation so difficult. Brines, chemical gradients, and ancient groundwater systems may once have persisted there, and perhaps in limited places still do. But this possibility, while scientifically serious, remains just that, a possibility. Mars continues to offer plausible niches without yet confirming occupation, and that is the great lesson Mars teaches. A planet can show ancient rivers, lake beds, minerals formed in water, organics in rock, and even puzzling atmospheric signals, and still not provide decisive proof that life was ever there. This is not failure. It is what a difficult scientific problem looks like when treated honestly. Mars demonstrates that life detection is strongest when independent lines of evidence converge. A single dramatic result may attract headlines, but it rarely settles the matter. What scientists truly need is agreement between geology, chemistry, isotopes, environmental context, and ideally repeatable observations across multiple instruments and samples. So Mars stands as both invitation and warning. It may once have been habitable, it may have preserved traces of ancient biology, it may still hide protected subsurface environments. But more than anything, it shows that the path from interesting chemistry to convincing biology is narrow and demanding. If life beyond Earth is ever confirmed, it will not be because one flashy clue briefly stole the spotlight. It will be because many different clues, gathered carefully and tested relentlessly, all ended up telling the same story. Europa changes the mood of the search for life. Mars is a world of ancient riverbeds, exposed rock, and the lingering question of whether something once lived there and vanished. Europa is different. Europa asks whether life might exist right now, in a place that sunlight never reaches, hidden beneath an outer shell of ice. It is a moon of Jupiter, slightly smaller than Earth's own moon, and at first glance it does not look especially generous. Its surface is bright, frozen, cracked, and brutally cold. But the scientific fascination comes from what may lie underneath. The most important idea about Europa is simple, though the consequences are enormous. Beneath that ice shell, scientists think there is a global ocean of liquid water. Not a few isolated pockets, not some damp slush in scattered cracks, but an ocean that may contain more water than all of Earth's oceans combined. If that is true, then Europa is not just an icy moon. It is one of the most significant potential habitats in the solar system. This hidden ocean is not visible directly, so it has to be inferred from several lines of evidence. One major source was the Galileo mission, which orbited Jupiter in the 1990s and early 2000s. Galileo studied Europa's gravity, surface features, and magnetic environment. The moon's surface is laced with long fractures, ridges, disrupted terrains, and regions where the ice appears to have shifted, broken, and in some cases refrozen. These patterns suggest a crust that is mechanically active rather than completely rigid. It looks less like a dead frozen shell, and more like ice interacting with something mobile beneath it. Magnetic field data provided another powerful clue. Jupiter has a strong magnetic field, and as Europa moves through it, changes in the surrounding magnetic environment can induce secondary magnetic effects inside the moon. The measurements fit well with the presence of a conductive layer beneath the surface, most likely a salty liquid ocean. That does not prove every detail, but it adds to the case with impressive weight. Gravity measurements also help. Europa's internal structure appears consistent with planetary differentiation, meaning the Moon separated into layers early in its history, with denser material sinking inward and lighter material remaining above. In this picture, Europa likely contains a metallic core, a rocky interior, and above that a deep water layer beneath the icy crust. Then there is heat. This is where tidal heating enters the story. Europa is constantly pulled and flexed by Jupiter's immense gravity, as well as by gravitational interactions with neighbouring moons. That repeated squeezing and stretching generates internal heat. The Moon is in effect being kneaded by gravity. It is not the sort of spa treatment most worlds would request, but it is scientifically very useful. Without tidal heating, Europa's water would be far more likely to remain frozen solid. With it, liquid water can persist beneath the surface over immense stretches of time. That matters because habitability is not only about water, it is about water, chemistry, and energy existing together for long enough to matter. If Europa has a global ocean, a rocky seafloor, and internal heating, then it may also have chemical gradients similar in principle to those found around hydrothermal systems on Earth. This is one of the reasons Europa became so important to ocean world science. On Earth, life does not depend exclusively on sunlight. Entire deep ocean ecosystems survive through chemosynthesis, drawing energy from chemical reactions rather than from photosynthesis. Microbes near hydrothermal vents use compounds such as hydrogen sulfide and other reduced chemicals to power metabolism, and larger organisms ultimately depend on those microbial foundations. If Earth can support life in dark seafloor environments, then sunlight stops looking like a requirement and starts looking more like one successful option among several. That shift is crucial. Europa forces scientists to broaden their picture of what a living world can be. A habitable environment does not have to resemble a planet with blue skies, continents, and sunlight falling through leaves. It can be a dark ocean beneath miles of ice, heated from within, chemically active, and shielded from the harsh radiation of space by its own frozen outer shell. In some ways, that is even more intellectually unsettling than finding a second Earth. A second Earth would confirm that familiar conditions can host life elsewhere. Europa raises the possibility that life may thrive in environments humans once would have ignored completely. Of course, the word may is doing plenty of work there. Europa is scientifically compelling, but it is not a confirmed living world. The thickness of its ice shell is still debated. The details of how material moves between the surface and the subsurface ocean remain uncertain. The nature of the seafloor, the availability of biologically useful chemistry, and the long-term stability of possible habitats all remain open questions. There is good evidence for a hidden ocean. There is not yet evidence for organisms swimming about beneath the ice, filing marine paperwork, and getting on with their European affairs. That is why missions matter so much. NASA's Europa Clipper is designed to examine the Moon in far greater detail, studying its ice shell, surface composition, possible plume activity, internal structure, and the degree to which the ocean may interact with the rocky interior below. Those details matter because a water ocean on its own is not enough. The strongest case for habitability comes when water is paired with energy sources, an ongoing exchange of useful chemicals. If oxidants produced at the icy surface can move downward, and reduced compounds from the interior can move upward, then Europa may possess the sort of chemical imbalance that life can exploit. Europa also sits in a particularly harsh part of the Jovian system. Jupiter's radiation environment is severe, and the surface of Europa is constantly bombarded. Paradoxically, that may help as well as harm. Radiation can alter surface chemistry, producing oxidants and other reactive compounds that may become relevant if they are transported downward. At the same time, it makes the surface itself a poor place for exposed biology to persist. That is another reason the hidden ocean remains the true focus. Europa is not a place where scientists expect forests, fish, or anything theatrically visible. It is a place where microbial life, if present, would likely exist in darkness, under pressure, and far below the frozen crust. That possibility gives Europa a special place in the search for life, because it tests the deeper logic of the whole question. If scientists can identify a world with liquid water, internal heat, chemical energy, and a stable environment beneath the surface, then habitability stops being a narrow planetary category and becomes something wider, more flexible, and more difficult to dismiss. Life, if it exists there, would not be basking in sunlight. It would be proving that biology can persist in hidden oceans warmed by gravity and chemistry alone. And Europa is not the only world making that argument. It is one of the most famous, perhaps the most elegant, but not the only one. Elsewhere in the solar system there are other ocean worlds, and some of them may offer an even more practical advantage. Europa hides its ocean beneath a stubborn shell of ice. Other moons may actually send material from their interior out into space, where spacecraft can sample it far more directly. In the search for life, that is the sort of convenience scientists try not to call exciting too early, even when it very clearly is. Enceladus is the sort of moon that makes planetary scientists try very hard to remain calm in public. It is small, bright, icy, and orbiting Saturn at a distance where, by older expectations, it should have been little more than a frozen object minding its own business. Instead, it turned out to be one of the most compelling places in the solar system to search for life. The reason is simple and extraordinary. Enceladus appears to be spraying material from a subsurface ocean directly into space. That changes everything. Most ocean worlds make life difficult for spacecraft before they even arrive. If the water is buried beneath miles of ice, then any serious investigation seems to require drilling, melting, tunneling, or some other heroic engineering scheme that sounds excellent until someone has to build it. Enceladus offers a much more generous arrangement. From fractures near its south pole, often called tiger stripes, plumes of water vapour, ice grains, salts, and organic material erupt into space. That means a spacecraft can fly through material that appears to have come from the interior without digging through kilometres of crust. In planetary science, this is very close to a gift. Much of what is known about Enceladus comes from the Cassini mission, a joint effort involving NASA, the European Space Agency, and the Italian Space Agency. Cassini spent years exploring the Saturn system and revealed that Enceladus was not a dead frozen moon at all. Its south polar region was active, the plumes were real, their composition could be measured, their behaviour implied an internal source connected to liquid water. Over time, the evidence built toward a picture that is now difficult to ignore. Beneath Enceladus lies a global or near-global subsurface ocean, capped by ice, but still interacting with the rocky core below. That interior interaction is one of the most important parts of the story. Cassini found signs that the plume material contained salts and silica particles, consistent with water having interacted with rock at elevated temperatures. In plainer terms, the ocean does not seem to be an isolated, frozen reservoir. It appears to be geochemically active. That matters because life, at least as it is understood on Earth, needs more than water alone. It needs energy and access to useful chemistry. If water is circulating through a rocky interior and picking up minerals, dissolved compounds and heat, then Enceladus begins to look much more like a serious habitat and much less like a decorative snowball. One especially intriguing discovery was molecular hydrogen. On Earth, hydrogen can be a valuable energy source for microbes. In hydrothermal vent environments on the ocean floor, certain microorganisms use hydrogen in metabolic reactions, drawing energy from chemistry rather than from sunlight. This broader process is called chemosynthesis. It supports entire ecosystems in dark places where photosynthesis is impossible. The discovery of hydrogen in Enceladus plume material therefore mattered a great deal, because it suggested that water rock reactions on the seafloor might be creating chemical energy that microbes could, in principle, exploit. This is where analogies with Earth become genuinely useful rather than merely convenient. Deep ocean hydrothermal vent systems on Earth host life in total darkness. There, hot water emerges from the crust carrying reduced chemicals, and microbes use those chemical gradients to power metabolism. Larger organisms depend on that microbial foundation. Enceladus is not Earth, and no one should casually paste an entire vent ecosystem onto a moon orbiting Saturn. But the comparison matters because it shows that sunlight is not a universal requirement for life. If liquid water, organics, and chemical energy come together in a stable environment, then biology becomes at least plausible. Cassini also found simple and more complex carbon-bearing compounds in the plume material. That is important, because carbon chemistry lies at the centre of known life. Organic compounds can serve as building blocks, intermediates, or byproducts of biological systems. But this is exactly where caution is required. Rich organic chemistry is not the same as life. Carbon compounds can be produced through non-biological processes in watery environments, in hydrothermal systems, and even in interstellar space. A moon can be chemically interesting without being biologically inhabited. In fact, the universe appears to be very good at making organic molecules without consulting biology at all. So Enceladus sits in a fascinating position. It has water. It appears to have internal heat. It shows evidence of rock-water interaction. It contains organics. It may offer chemical energy in the form of hydrogen. All of that makes it one of the best astrobiological targets in the solar system. Yet none of it, by itself, proves that anything is alive there. A living ocean and a habitable ocean are not the same thing. The difference between them is the difference between possibility and evidence. That is why Enceladus sharpens the whole question into something practical and precise. If a spacecraft flies through a plume and detects intriguing chemistry, what exactly would count as a serious sign of life? A few organic molecules would not be enough. Salts and water would not be enough. Even hydrogen would not be enough. Scientists would need patterns that are difficult to explain through ordinary geochemistry alone. They might look for unusual distributions of organic compounds, strong chemical disequilibrium, isotopic ratios that suggest biological processing, or combinations of molecules that on Earth are strongly associated with metabolism. Ideally, they would want several lines of evidence at once, all pointing in the same direction and resisting non-biological explanations. This is what makes Enceladus so powerful in the wider search. It turns the abstract question of alien life into an engineering and measurement problem. The issue is no longer just where life might exist, but how a probe could detect the difference between a chemically rich ocean and a genuinely living one. Enceladus does not merely invite hope, it demands standards, and that may be its greatest value. It is one thing to imagine hidden life in a distant sea. It is another to fly through that sea's ejected spray and ask, with scientific discipline, what pattern in this material would survive skepticism. If life exists there, it may already be casting clues into space. The challenge is making sure those clues are read honestly, without mistaking interesting chemistry for biology, and without overlooking biology because it first arrived dressed as chemistry. Titan is valuable precisely because it does not behave like the kind of place people instinctively imagine when they think about life. Mars invites comparison with deserts and dried lake beds. Europa and Enceladus invite thoughts of dark oceans beneath ice. Titan does something more unsettling. It asks whether the search for life has been too tightly built around Earth from the beginning. Saturn's largest moon has a thick nitrogen-rich atmosphere, clouds, rain, rivers, lakes and seas. But the liquids on its surface are not water. They are hydrocarbons, mainly methane and ethane. Titan is also the only world besides Earth known to have stable liquids on its surface, and Cassini-Huygens showed that it has both that dense atmosphere and widespread methane and ethane lakes and seas. That makes Titan both familiar and deeply strange. In some ways it resembles Earth more than almost any other moon. It has weather, seasons, clouds, rainfall, channels carved by flowing liquid, and broad surface reservoirs. Yet the environmental conditions are radically different. Titan's surface is so cold that water ice behaves like bedrock, while methane can evaporate, condense, and fall as rain. What looks superficially like an Earth-like hydrological cycle is actually a hydrocarbon cycle. The result is a world that seems to echo Earth while quietly replacing nearly all the important ingredients. This is why Titan matters so much to the broader question of alien life. It may not be the most likely place for life as humans know it. If scientists were choosing a nearby world for ordinary water-based microbial biology, they would usually look first toward Mars, Europa, or Enceladus. But Titan forces a harder and more useful question. Could biology somewhere in the universe make use of solvents, chemical pathways, or environmental conditions that are very different from those on Earth? The Moon's atmosphere and surface are rich in complex organic chemistry, and that is scientifically important even before life enters the discussion. Cassini-Huygens revealed an atmosphere and surface environment full of prebiotic chemical potential, while NASA describes Dragonfly's goals in part around investigating Titan's habitability and the chemistry connected to life's origins. That phrase, prebiotic chemistry, matters here. It refers to chemistry that may precede life, rather than prove life. Titan is filled with carbon-bearing compounds, produced by reactions high in the atmosphere and deposited onto the surface. Sunlight and energetic particles drive reactions in the upper air, building increasingly complex molecules from nitrogen and methane. Some fall as haze, some dissolve in surface liquids, and some may accumulate across long spans of time. This does not mean Titan is alive. It means Titan is chemically active in ways that may help scientists understand how far non-living chemistry can go before biology becomes necessary. There is another reason Titan remains so compelling. Beneath its icy crust, Cassini-Huygens findings also support the existence of a subsurface ocean, likely made of water mixed with ammonia. That means Titan may host two very different chemically interesting realms at once: a frigid hydrocarbon-rich surface and a deeper internal water ocean. If there is any place in the solar system that politely refuses to fit into one neat category, it is Titan. It is not just a frozen moon. It is a layered world with atmospheric chemistry, surface lakes, organic haze, water ice, and likely internal liquid water. This is where the speculation about weird life enters, and where it must be handled carefully. Some researchers have explored whether extremely exotic biochemistry could, in principle, function in liquid methane or ethane. Those discussions are scientifically useful because they test how much of biology is truly universal and how much is an accident of Earth's conditions. But Titan is not evidence that methane-based life exists. It is a reminder that detection methods should not be so narrow that they only recognize something that already looks Earth-like. A search built entirely around oxygen, liquid water at the surface, and familiar biological byproducts might miss a world that is chemically active in more unusual ways. That does not mean scientists should become wildly permissive and start calling every strange molecule a possible alien fish. Restraint matters even more on Titan than elsewhere. The Moon is a lesson in how easy it is to be seduced by novelty. Strange does not mean alive. Rich organic chemistry does not mean biology. Unusual solvents do not automatically imply unfamiliar organisms making the best of them. Titan's scientific value lies partly in forcing researchers to ask better questions, not in providing answers too early. The Cassini-Huygens mission made that possible by transforming Titan from a hazy orange mystery into a place with mapped lakes, atmospheric profiles, surface images, and a much clearer chemical story. Huygens descended through the atmosphere and reached the surface. Cassini revealed the lakes and seas, the methane cycle, and the broader complexity of the Moon. Now NASA's Dragonfly mission is intended to build on that legacy. NASA describes Dragonfly as a rotorcraft mission to Titan that will investigate the Moon's habitability, sample material at multiple sites, and study chemistry tied to the origins of life, with launch currently set no earlier than 2028. Dragonfly is especially fitting for Titan because Titan's dense atmosphere and low gravity make flying unusually practical there. A rover would be limited. A flying laboratory can visit several sites and compare different chemical environments. That matters because Titan is not simply one setting. It is an entire system of dunes, icy materials, atmospheric deposits, and organic-rich landscapes. The mission is not being sent to confirm exotic life. It is being sent to understand habitability, prebiotic chemistry, and how far complex carbon chemistry can develop on a world that is both alien and oddly Earth-like. And that wider lesson becomes even more important once the search moves beyond the solar system. Nearby worlds can be visited, sampled and studied in detail. Distant worlds usually cannot. There, scientists will often have only faint atmospheric signals, reflected light, and limited context. Titan is therefore more than a curious moon. It is a warning against lazy assumptions. If life elsewhere is genuinely unfamiliar, then the hardest part may not be finding a strange world. It may be learning not to mistake unfamiliarity for impossibility. A planet can be interesting without being habitable, and it can be habitable without being inhabited. That distinction sits near the centre of astrobiology. When scientists judge whether a world might support life, they are not asking only whether it is rocky or whether it vaguely resembles Earth in an artist's rendering. They are asking whether the planet can maintain the conditions needed for biology over meaningful spans of time. That usually means liquid water, access to useful chemistry, an energy source, enough gravity to help retain an atmosphere, and some degree of long-term environmental stability. A brief warm period or a thin film of water appearing for a geological moment is not the same thing as a durable habitat. Liquid water remains such a central part of this discussion because it is the only solvent known to support life, and because it is chemically excellent at enabling reactions, transport, and complexity. But water alone is not enough. A world may have water and still be sterile if it lacks energy gradients, nutrient cycling, or environmental persistence. Scientists therefore think in systems. They look at atmosphere, geology, internal heat, surface conditions, and the interaction between star and planet. James Lovelock's broader planetary thinking, especially in the context of Gaia, helped popularize the idea that a living world is not just a collection of organisms, but a whole interacting system of air, rock, water, and chemistry. Even without adopting every strong interpretation of Gaia, planetary science still relies on this systems view. That is one reason the habitable zone is useful but limited. In the simplest sense, the habitable zone is the range of distances around a star where a planet with the right atmosphere could allow liquid water to exist at the surface. It is sometimes called the Goldilocks region, which is charming but also slightly dangerous because it makes the whole thing sound tidier than it is. Being in the habitable zone does not guarantee oceans, weather stability, or life. It only means that surface liquid water is not ruled out immediately by distance from the star alone. Atmospheric greenhouse effects, cloud feedbacks, surface reflectivity, planetary mass, and geochemical cycling all matter enormously. ESA notes this directly. A planet's real temperature depends strongly on its atmosphere and greenhouse effect, not just its orbital distance. The greenhouse effect is especially important because it is not some optional climatic accessory. It is one of the main reasons Earth is habitable at all. Without the natural greenhouse effect, ESA notes that Earth's average temperature would be about 30 degrees Celsius lower than it is now. That is a reminder that a planet's atmosphere is not just decorative sky. It traps heat, redistributes energy, and helps determine whether water remains liquid or disappears into ice. Too little greenhouse warming and a planet may freeze. Too much, and it may shift toward conditions more like Venus. So when scientists discuss habitability, they're often really discussing climate regulation, whether a world can hold onto an atmosphere, maintain temperate conditions, and avoid running too far toward either extreme. Long-term stability matters because life, as far as anyone knows, is not fond of constantly being reset. Biology requires time. It needs environments that persist long enough for chemistry to become organized, for metabolism to emerge, and for evolution to do its slow, patient work. That is why geochemical cycling becomes so important. Carbon cycling, water cycling, volcanic outgassing, weathering, and atmosphere surface interactions all help determine whether a planet remains clement or drifts into hostility. A planet that is habitable for a few million years is far less promising than one that stays broadly stable for hundreds of millions or billions of years. The question is not only, could life survive there today, but also, could life have had time to begin and persist there at all? The host star also matters far more than older public discussion sometimes implied. A planet is not habitable in isolation. It lives inside the behavior of its star. The amount of light and heat a star provides shapes the location of the habitable zone, but the star's activity matters too. ESA notes that the habitable zone around a small red dwarf star lies much closer to the star than the habitable zone around the Sun, and that such stars can remain active for far longer, exposing close-in planets to more stellar outbursts. That becomes a major concern because the worlds easiest to detect are often orbiting these small stars. Red dwarfs, also called M-dwarfs, are scientifically important because they are common and because rocky planets in their habitable zones are easier to find and study. But they also create headaches. NASA astrobiology notes that planets in these close-in habitable zones are often tidally locked, with one side always facing the star, and that young red dwarfs can produce powerful flares. NASA has also highlighted how an active red dwarf like Proxima Centauri can threaten atmospheric survival through intense high-energy radiation that strips gases away. In other words, the same stars that give astronomers convenient targets may also produce harsh environments that complicate habitability. Ultraviolet radiation and flare activity matter not only for life itself, but also for detectability. Strong stellar activity can alter atmospheric chemistry, mimic or erase potential biosignatures, and make a planet's spectral fingerprints harder to interpret. NASA's astrobiology strategy explicitly describes ultraviolet flare measurements around low-mass stars as crucial for assessing atmospheric retention, planetary habitability, and the interpretation of biosignatures. This means the star is part of the measurement problem as much as the planet is. A world might look chemically unusual not because life is shaping it, but because its star is blasting the atmosphere into odd states. Tidal locking adds another layer of complexity. At first glance, a planet with permanent day on one side and permanent night on the other sounds grim, as though one hemisphere is forever roasting while the other freezes in silence. But climate modelling has shown that this does not automatically make such planets uninhabitable. Much depends on the thickness of the atmosphere and the planet's ability to move heat around. NASA's exoplanet strategy cites climate studies of synchronously rotating planets because the basic picture is more subtle than simple extremes. A tidally locked planet might still maintain habitable regions if its atmosphere and perhaps its oceans redistribute energy efficiently enough, so even here habitability is not a yes or no button. It is a climate question. The idea of the habitable zone also becomes less rigid once internal oceans and protected niches enter the picture. Worlds outside the classic surface habitable zone may still host liquid water beneath ice if internal heat is available, as with ocean worlds in the outer solar system. That means a planet or moon can be far too cold for surface oceans and still remain astrobiologically interesting. Habitability, then, is broader than a ring around a star. It can include subsurface oceans, buried hydrothermal systems, and localized refuges protected from surface extremes. That shift is important because it prevents the search from becoming too narrow. A world does not have to look Earth-like from the outside to be worth taking seriously. So when scientists judge whether a planet is habitable, they're really assembling a long checklist of interacting conditions. Is there likely liquid water, either at the surface or below it? Is there an atmosphere, and can it survive? Are their greenhouse effects strong enough to warm the planet, but not so strong that they destroy clement conditions? Is there a reliable energy source? Is chemistry being cycled through the environment in useful ways? Is the host star relatively stable, or is it busy trying to strip the atmosphere off every nearby world? And has the environment persisted long enough for life to do more than merely flirt with possibility? Once scientists identify planets that seem potentially habitable by those standards, a new difficulty begins. Most of those worlds are far too distant to visit. No rover is driving there. No probe is scooping up sediment. The next question becomes how anyone can study such planets from immense distances, using only tiny changes in starlight, faint atmospheric signatures, and the delicate art of reading chemistry from light alone. Before, a gas giant roasting close to a star may be fascinating in its own dramatic way, but it is not usually the place people begin when thinking about biology. Detection comes first because it allows the search to narrow from endless possibility into a list of actual worlds with measurable properties. The most productive method for finding exoplanets has been the transit method. This works when a planet passes in front of its star from the point of view of the observer. As it crosses the star's face, the star becomes very slightly dimmer. That dip in brightness can be measured. If it repeats at regular intervals, it suggests that a planet is orbiting the star and blocking a tiny fraction of the light each time it passes. The amount of dimming helps astronomers estimate the planet's size, especially its radius. A larger planet blocks more light. A smaller planet blocks less. The timing between transits reveals the orbital period, which helps show how close the planet is to its star. This method has been incredibly powerful because it can be applied to huge numbers of stars at once. The Kepler Space Telescope used it to transform exoplanet science, showing that planets are not rare oddities, but a normal feature of the galaxy. Kepler stared at one patch of sky and watched for repeating dips in starlight, discovering thousands of candidate planets and confirming that small rocky worlds are common. The Transiting Exoplanet Survey Satellite, often called TESS, continued that work by surveying much more of the sky and focusing on stars closer to Earth, making follow-up observations easier. These missions did not just add planets to a catalogue. They changed the scale of the question. The universe went from possibly containing planets to being visibly crowded with them. The transit method does have limits. It only works when the orbital geometry lines up properly. Most planetary systems are tilted at angles that do not produce visible transits from Earth. So even if a star has planets, the transit method may miss them completely. It also tells astronomers more readily about size than about mass. A planet's radius is useful, but it does not reveal what the planet is made of. A large radius could mean a gas giant, a swollen hot world, or a lower density planet with a thick atmosphere. To understand composition, another method becomes crucial. That method is radial velocity. Instead of watching a star dim, astronomers watch the star wobble. A planet does not orbit while the star remains perfectly still. Both objects orbit their shared centre of mass. Because the star is so much more massive, its movement is smaller, but it is still there. As the star moves slightly toward and away from Earth, its light shifts in wavelength through the Doppler effect. By measuring those shifts very precisely, astronomers can infer the presence of an orbiting planet and estimate a minimum mass for it. This technique was central to the first confirmed discovery of a planet orbiting a Sun-like star, made by Michel Mayor and Didier Queloz in the 1990s. Radial velocity is especially important because when it is combined with transit data, astronomers can estimate density. Radius from transits plus mass from radial velocity gives a much clearer picture of what kind of world is being observed. A dense planet is more likely to be rocky. A low-density planet is more likely to be gaseous or rich in volatiles. This is one of the key steps in narrowing the search. Scientists are not just finding planets, they are sorting them into rough physical types. Some are hot Jupiters, huge gas giants orbiting absurdly close to their stars. Some are mini Neptunes, smaller but still wrapped in thick atmospheres. Some are rocky worlds more similar in size to Earth or Venus. Detection becomes classification, and classification determines what is worth examining more closely. Other methods help as well, though they are less commonly the main source of potentially habitable targets. Direct imaging attempts to photograph planets by blocking out the light of the host star, which is technically very difficult, but extremely valuable when it works. Gravitational microlensing takes advantage of gravity-bending light, allowing astronomers to detect planets when a foreground star system briefly magnifies the light of a more distant background star. This method can reveal planets that other techniques miss, including worlds farther from their stars. There is also astrometry, which measures the precise motion of stars across the sky, though this has historically been more challenging for planet hunting. Each method has strengths, weaknesses, and biases, which means the known population of exoplanets is shaped partly by what current instruments are best at finding. That bias matters. Large planets close to stars are easier to detect than small planets farther out. Worlds orbiting small stars often produce clearer signals than worlds orbiting large bright ones. This means astronomers do not begin with a fair sample of all planets everywhere. They begin with the planets most detectable by existing methods. Even so, that is enough to reveal an astonishing variety of worlds. Once a planet is found, several measurements become immediately important. Radius helps show size. Mass helps reveal how much matter is present. Density suggests whether the planet is likely rocky, watery, or gaseous. Orbital period indicates distance from the star. Stellar type matters because a planet around a cool red dwarf receives energy differently from one orbiting a sun-like star. From those pieces, astronomers can begin asking whether the world might be hot, frozen, temperate, airless, bloated, dense, or potentially habitable. None of this proves life. It does something more basic and necessary. It tells scientists whether the planet deserves the scarce and valuable attention of deeper study. That is why detection comes first. Before chemistry, before atmospheres, before biosignatures, there has to be a real world with measurable properties and a plausible case for further interest. Only then can the search move from finding planets to examining them, and once a promising exoplanet has been identified, the next challenge becomes even stranger. Astronomers begin trying to read the chemistry of its atmosphere not by touching it, landing on it, or sampling it directly, but by studying the tiny fraction of starlight that has passed through the planet's air on its way across the galaxy. One of the strangest parts of the search for life is that astronomers often have to study a planet without ever seeing it as a clear world. No oceans are visible, no clouds can be watched drifting in detail, no lander is scooping up soil. Instead, they study light, tiny changes in light, faint distortions in light, missing pieces of light. From those small clues, they try to work out what an atmosphere is made of on a world that may be dozens or hundreds of light years away. It is an absurdly delicate business, and yet it is one of the most powerful tools in the entire search. The key method is spectroscopy. At its heart, spectroscopy rests on a simple idea. Atoms and molecules do not interact with light randomly. Each one absorbs and emits light at particular wavelengths, depending on its structure. In effect, every substance leaves a kind of pattern, not a neat little signature written in cursive, sadly, but a distinct set of spectral features that can be recognized if the measurements are good enough. If light passes through a planetary atmosphere, some wavelengths are absorbed more strongly than others. By studying which parts of the light are missing, astronomers can infer which molecules may be present in that atmosphere. This is remarkable because it means a planet does not need to be visited in order for its chemistry to begin revealing itself. Its atmosphere, if it has one, can alter the starlight passing through it. The challenge is that the effect is very small. The planet itself is tiny compared with the star, and the atmospheric contribution is smaller still. So the signal astronomers want is often hidden inside noise, instrumental limitations, and the general fact that the universe does not seem especially interested in making this easy. One important approach is transit spectroscopy. This works when a planet passes in front of its star. During the transit, a very small portion of the starlight filters through the outer edge of the planet's atmosphere before reaching the telescope. Molecules in that atmosphere absorb particular wavelengths, leaving subtle features in the light. By comparing the star's spectrum during the transit with the spectrum when the planet is not crossing, astronomers can begin to identify atmospheric ingredients. This is one reason transiting planets are so valuable. They provide not only a way to detect the planet, but also a chance to probe the chemistry of its air. Another technique is emission spectroscopy. In this case, astronomers study light coming from the planet itself, usually in the infrared, where warm objects emit radiation. When a planet moves behind its star, the total light from the system drops slightly because the planet's contribution disappears for a time. By comparing the light before and during that hidden phase, astronomers can estimate what the planet itself was adding. That can reveal information about temperature and atmospheric composition, especially for hotter planets, where the emitted signal is stronger. It is less like reading a detailed weather report and more like trying to deduce the existence of soup by measuring warmth through a wall. But it is still scientifically powerful. There is also reflected light spectroscopy. Here, the goal is to study starlight that has bounced off the planet's atmosphere or surface. This is extremely difficult because stars are so much brighter than their planets, but it matters because reflected light can contain information about clouds, hazes, atmospheric gases, and even surface properties. Direct imaging efforts often connect with this approach, especially when astronomers try to isolate a planet's faint glow from the overpowering glare of its star. These methods are supported by some of the most advanced observatories humans have built. The Hubble Space Telescope helped open this field by studying exoplanet atmospheres long before it was fashionable to speak confidently about them. The James Webb Space Telescope has taken that work much further, especially in the infrared, where many important molecules leave strong features. Major ground-based observatories also contribute using large mirrors, precise spectrographs, and careful calibration to extract signals that would once have been lost entirely. Future observatories are being designed with atmospheric characterization as one of their central goals, because the search for life depends not just on finding planets, but on reading the chemistry of those planets in far greater detail. So what are scientists actually looking for in these atmospheres? Water vapor is one obvious target, because water matters to habitability and leaves identifiable spectral features. Carbon dioxide is important because it affects climate, greenhouse warming, and atmospheric structure. Methane attracts attention because it can be associated with biology, though it can also be produced geologically. Oxygen, which gases are present together, in what proportions, under what stellar conditions, in what temperature regime, with what likely surface or ocean interactions. Does the chemistry appear close to equilibrium, or does it seem persistently out of balance? An atmosphere that contains one intriguing molecule may simply be interesting. An atmosphere containing combinations of gases that should not remain together for long, without constant replenishment, becomes much more persuasive. This is where life detection begins to look less like treasure hunting and more like careful forensic science. And the data are often difficult to interpret. Signals can be weak. Clouds and hazes can obscure deeper atmospheric layers. Stellar activity can contaminate the spectrum. Different molecules can mimic each other in overlapping wavelength regions. Instrument noise can distort what is really there. Sometimes the result is not a crisp identification, but a range of possibilities that must be tested against models. Astronomers therefore spend a great deal of time not only observing, but arguing productively with the data, comparing models, checking alternative explanations, and trying very hard not to fall in love with their favourite interpretation too soon. This is where the central logic of the search becomes very clear. life detection at interstellar distances is not going to come from one molecule floating heroically through a telescope's field of view. It will come from an atmospheric pattern that keeps looking unusual in biologically suggestive ways, even after every obvious non-biological explanation has had its chance. The search becomes one of chemical disequilibrium, environmental context, and repeated pattern recognition. In other words, astronomers are not simply reading atmospheres. They are trying to decide whether the chemistry of a distant world looks as though something active, organized, and persistent may be shaping it from within. Of all the gases astronomers might hope to find in a distant atmosphere, oxygen has long held a special place. That is not because oxygen is rare in the universe as an element. It is not. Oxygen is common in rocks, water, and many minerals. What makes atmospheric oxygen so interesting is the difficulty of keeping large amounts of it floating freely in the air for long periods without some process constantly replacing it. On Earth, that process is life. More specifically, it is photosynthesis, carried out first by ancient microbes and later by plants, algae, and other organisms that learned how to use sunlight to split water and build sugars, releasing oxygen as a byproduct. That is why oxygen became one of the classic biosignature candidates. A planet with a lot of oxygen in its atmosphere starts to look, at least from a distance, like a world where biology may be doing serious work. Ozone matters for a related reason. Ozone is made from oxygen, and in an atmosphere rich in oxygen, some of that oxygen can be rearranged into ozone by ultraviolet radiation. Ozone does not replace oxygen as a biosignature candidate, but it can act as a useful stand-in, especially because it has strong spectral features that may sometimes be easier to detect. In that sense, oxygen and ozone often travel together in astrobiology. One is the main atmospheric gas of interest, and the other can help reveal its presence indirectly. Earth is the reason these gases became so important. The atmosphere today contains a great deal of oxygen, and that oxygen is not there by accident. It is the result of life changing the planet over immense stretches of time. But Earth also provides the warning label. Even a genuinely living planet may not always advertise itself with oxygen. For a very large part of Earth's early history, life already existed while atmospheric oxygen remained low. Microbes lived, evolved, and altered local environments long before the air became rich in oxygen. That means a living world can be alive in every meaningful sense, and still fail to announce itself in the most obvious way astronomers might hope for. This long delay is tied to one of the most important transitions in Earth's history, the Great Oxidation Event. Before that event, Earth's atmosphere contained very little free oxygen, yet life was already here. Cyanobacteria, those ancient microbial pioneers, had developed oxygenic photosynthesis and were releasing oxygen into the environment. The problem was that the planet had many sinks for oxygen. Iron in the oceans, reduced volcanic gases, and other reactive materials absorbed it. Only after those sinks were increasingly filled or overwhelmed did oxygen begin to accumulate more substantially in the atmosphere. So Earth's history shows something crucial. A planet may host life for a very long time before oxygen becomes a strong atmospheric signal. That has major consequences for the search for life elsewhere. If astronomers treat oxygen as the one great answer, they may miss planets that are biologically active, but still in an earlier stage of atmospheric development. A distant world could be full of microbes, biochemistry, and evolutionary potential while looking disappointingly unoxygenated from afar. In other words, oxygen is exciting, but its absence does not automatically mean lifelessness. Life may be present and simply not yet loud enough. Even when oxygen is present, the situation is not as simple as it first appears. Modern astrobiology teams, including researchers working through NASA-linked programs and university biosignature modelling groups, spend a great deal of effort studying how oxygen and ozone can be produced without life. These are called abiotic routes, and they matter because a false positive in life detection would be a rather unfortunate way to make history. One important abiotic mechanism is photodissociation, This happens when high-energy ultraviolet light breaks apart molecules such as water or carbon dioxide in the upper atmosphere. If hydrogen escapes to space more easily than oxygen, oxygen can build up behind it. Under certain stellar and atmospheric conditions, that build-up could become substantial. Atmospheric escape is therefore part of the story as well. On some planets, especially around active stars, lighter elements can be stripped away over time. If a planet loses enough hydrogen from broken apart water, it can be left with an oxygen-rich atmosphere that looks intriguing but has no biology behind it. Ozone may then form from that oxygen, making the atmospheric picture even more suggestive. A telescope might detect oxygen or ozone and see what looks like a promising world, when in fact the atmosphere is the scar left by water loss and harsh radiation rather than the breath of a biosphere. That is why context matters so much. Astronomers do not want to know only whether oxygen is there. They want to know what kind of star the planet orbits, how active that star is, what other gases are present, whether the planet shows signs of water loss, whether the atmosphere appears stable, and whether the chemistry makes sense as a whole. Oxygen by itself is interesting. Oxygen in the wrong context can be misleading. Oxygen in the right context becomes more compelling, especially when it appears alongside other gases that are difficult to explain through non-biological processes alone. This is where atmospheric disequilibrium becomes especially useful. On Earth, oxygen does not merely sit politely in the sky as an isolated fact. It exists alongside methane and other gases in ways that are chemically revealing. Oxygen is highly reactive. If large amounts of it persist, something usually has to be replenishing it. If methane also persists at the same time, despite the fact that oxygen tends to destroy it, the atmosphere begins to look even more unusual. That coexistence suggests active sources are constantly feeding the system. On Earth, those sources are strongly tied to biology, so oxygen remains a classic biosignature candidate for good reasons. It is tied to one of the most transformative biological processes known, photosynthesis. It helped reshape Earth on a planetary scale. Through ozone, it may also reveal itself in detectable ways. But Earth itself teaches that oxygen is not a universal clock that starts ticking the moment life appears. A living planet may spend vast spans of time without much oxygen in the air. And planetary physics teaches that oxygen can also arise through non-living pathways if the conditions are right. That leaves oxygen in a very interesting position. It is not the answer by itself, but it is far too important to ignore. It is best understood as one major piece of a larger atmospheric puzzle. If oxygen appears alongside methane, sulfur-bearing gases, water vapour, and other chemically unlikely partners, the case becomes more interesting. If several of those gases coexist in ways that should not persist naturally, then the atmosphere begins to tell a richer story than oxygen alone ever could. One of the most powerful ideas in the search for life is also one of the least dramatic sounding. It is called chemical disequilibrium. That phrase does not have the glamour of alien oceans or glowing exoplanets, but it may turn out to be one of the clearest ways to recognize a living world from far away. The basic idea is simple. Some mixtures of gases should not remain together for very long. Chemistry, left to itself, tends to move toward balance. Reactive substances combine, break down, and settle into more stable arrangements. So if an atmosphere stays in a persistently unstable-looking state, something may be actively maintaining it. That something does not have to be life. Volcanoes can inject gases, sunlight can drive photochemistry, geological processes can keep a system busy for a long time. But on Earth, one of the clearest examples of large-scale atmospheric disequilibrium is strongly tied to biology. The atmosphere contains abundant oxygen, and it also contains methane. Those two gases are not natural long-term companions. Oxygen is highly reactive and methane is chemically eager to be destroyed in an oxygen-rich atmosphere. If Earth were left without constant replenishment, methane would decline fairly quickly. Yet it remains present because biological and geological processes keep supplying it. The same is true of oxygen, which would also fade over geological timescales if life stopped replacing it. The key point is that both gases coexist in a way that requires active ongoing sources. This is why Earth matters so much as the reference world. It shows that a planet can become chemically strange because life is reshaping it from within. Earth's atmosphere is not just a passive shell resting above rocks and oceans. It is part of a global system involving biology, climate, oceans, geology, and sunlight. That broader planetary view connects with the work of James Lovelock, who argued that life can drive a planet far from simple chemical equilibrium. Lovelock's disequilibrium argument remains influential because it shifts the question from finding one miraculous gas to recognizing an atmosphere whose overall chemistry looks maintained in an improbable state. That shift is important because single gases are too easy to misunderstand. Oxygen alone can mislead. Methane alone can mislead. Water vapour tells astronomers something useful about conditions, but it is not itself a life detector. Disequilibrium is more powerful because it relies on relationships rather than isolated ingredients. It asks not just what is present, but what is present together, in what quantities, under what conditions, and for how long. A living world may not advertise itself by one chemical shout. It may reveal itself by quietly maintaining several incompatible things at once. Modern atmospheric modelling builds on exactly this logic. Researchers studying biosignatures do not simply search for one ideal gas, they test combinations. They ask how atmospheres behave under different stellar types, pressures, temperatures, surface chemistries, and radiation environments. They model what biology might produce, and then compare that with what geology, volcanism, photochemistry, and ocean-atmosphere interactions could also create. The work becomes less like collecting clues from a treasure map, and more like running a long planetary interrogation. Which chemical patterns survive questioning? Which collapse under alternative explanations? Which become stronger when combined with the rest of the environment? Methane and oxygen are the classic example, because Earth shows how revealing that pairing can be. But other gases matter too. Sulfur chemistry is especially interesting because sulfur-bearing compounds can be produced both biologically and geologically, and the balance between them may tell an important story. Sulfur dioxide, hydrogen sulfide, and related compounds can reflect volcanism, atmospheric processing, microbial metabolism, or combinations of these. On their own, they're usually ambiguous, but when read as part of a larger atmospheric pattern, they may strengthen or weaken the case for life. A chemically active planet may produce sulfur compounds naturally. A biosphere may alter their abundance, cycling, or seasonal behaviour in ways that become suggestive when paired with other clues. Seasonal variation is another piece of the puzzle. Life is not only matter, it is process. Processes create rhythms. On Earth, biological activity changes with seasons, temperature, sunlight, and ecosystem dynamics. This affects gases in the atmosphere, surface reflectance, and chemical cycling. If a distant world showed regular changes in atmospheric composition that repeated in a way tied to its orbit, temperature, or illumination, that would be interesting. Not decisive on its own, certainly, because non-biological cycles can also be seasonal. But repeated patterns can reveal active systems, and active systems are always worth taking seriously. A planet that breathes chemically, even faintly, becomes more compelling than one that presents only a single still image. Surface reflectance adds another layer. Different materials reflect light differently, and living systems can leave broad surface signatures. On Earth, vegetation alters the way land reflects light, especially near the edge of visible and infrared wavelengths. That does not mean astronomers expect to detect forests neatly outlined on a distant planet. The signals are much subtler than that. But in principle, large-scale biological pigments or repeating surface changes could contribute to a wider biosignature case. Once again, the value is strongest when combined with atmospheric evidence rather than treated as a lone miracle. This is why multi-phase observations matter so much. A convincing biosignature case is unlikely to come from one measurement taken once. Scientists would want to observe the planet at different points in its orbit, perhaps during transits, secondary eclipses, or direct observations if possible. They would want to compare atmospheric signals over time, test the stability of the interpretation, and look for links between chemistry, temperature, clouds, and perhaps even surface behaviour. The more independent pieces begin to align, the stronger the case becomes. A single odd gas may be curious. A whole planet showing coordinated chemical and physical patterns over time becomes far harder to dismiss. Even then, caution remains essential. Chemical disequilibrium is powerful, but it is not magical. Planets are not simple laboratory flasks. They are messy systems shaped by stars, geology, atmospheric escape, cloud layers, volcanism, impacts, and chemistry that may be more creative than human intuition prefers. An atmosphere can look odd for many reasons. This is why the strongest biosignature patterns are not merely unusual. They are unusual in ways that survive repeated attempts to explain them without life. That distinction is everything. The hardest part of life detection is not finding a signal that seems exciting. Nature can provide exciting signals very easily. The hardest part is ruling out the many non-biological processes that can imitate fragments of a living world. An atmosphere rich in oxygen might result from water loss. Methane might come from water-rock interactions. Sulfur compounds may reflect volcanic outgassing. Seasonal changes might arise from circulation, freezing, thawing, or photochemical cycles rather than biology. A good biosignature case therefore depends not only on what is found, but on what has been excluded. This is why disequilibrium is best understood as a framework rather than a shortcut. It guides scientists toward the kinds of patterns that life might maintain on a planetary scale. It rewards combinations, context, repeatability, and system-wide thinking. It also encourages humility, because even the strongest-looking pattern must still pass through every alternative explanation the planet can throw back. So the search does not end when astronomers find something chemically strange. In many ways, that is where the real work begins. A possible biosignature becomes meaningful only after geology, atmospheric physics, stellar activity, and photochemistry have all had the chance to explain it away and failed. Until then, the planet remains intriguing but unproven, and that is the central difficulty of the whole search. It is not just about finding atmospheres that look alive, it is about making sure they are not simply very good at pretending. If life detection is one of the most profound goals in science, then false positives are one of its most important disciplines. A false positive is not just an annoying mistake or an awkward footnote after an exciting headline. It is one of the central reasons the whole field must move carefully. Nature is perfectly capable of producing things that look suggestive, impressive, and suspiciously biological without any biology being involved at all. Volcanoes can release reactive gases. Ultraviolet radiation can reshape atmospheres. Water and rock can interact in surprisingly rich ways. Planets can lose one part of an atmosphere and be left with something misleading behind. In other words, the universe can fake parts of life's signature without ever becoming alive. That is why life detection is not simply a search for interesting chemistry. It is a search for chemistry that remains interesting after every serious non-biological explanation has been given the chance to explain it away. The real standard is not excitement. It is survival under scrutiny. Volcanoes are one of the oldest and most effective imposters in this story. Volcanic activity can inject gases such as carbon dioxide, sulfur dioxide, hydrogen, methane under some conditions, and many other chemically important compounds into an atmosphere. A geologically active world may therefore look dynamically alive even if nothing biological is present. Sulfur-bearing gases are especially tricky because they can participate in atmospheric chemistry, influence clouds, and interact with sunlight in complicated ways. On a distant planet, a volcanic atmosphere might produce combinations of gases that look unusual enough to attract attention. Without geological context, it would be easy to overinterpret them. Water-rock reactions create another major source of confusion. When water interacts with certain minerals, especially in hydrothermal systems, it can generate hydrogen and methane without any biology at all. This matters because hydrogen and methane are both chemically interesting gases, and methane in particular often draws attention because of its connection to life on Earth. But on another world, methane might be produced through serpentinization and related geochemical processes rather than metabolism. A world can therefore have water, chemical gradients, and organic production without crossing the line into biology. The chemistry may be rich, promising, and worth studying, while still being entirely non-living. Photochemistry complicates things even further. Sunlight, especially ultraviolet radiation, can drive atmospheric reactions that create or destroy molecules in ways that imitate biological patterns. Oxygen and ozone are classic examples. On Earth, abundant atmospheric oxygen is strongly associated with life, especially photosynthesis. But under the right conditions, ultraviolet radiation can split water vapour or carbon dioxide. And if lighter hydrogen escapes to space more easily than oxygen, oxygen can accumulate without biology ever lifting a finger. Ozone may then form from that oxygen, making the atmosphere look even more suggestive. So a distant oxygen-rich atmosphere may not be a biosphere. It may be the result of photodissociation, atmospheric escape, and a planet being chemically pushed around by its star. Atmospheric escape itself is a major actor in this story. If a planet is exposed to strong radiation, especially from an active young star or a close red dwarf, it may gradually lose lighter components of its atmosphere. Over long periods, that can reshape the planet's atmospheric composition dramatically. The result may be an oxygen-rich atmosphere that looks exciting from afar, but is really the aftermath of water loss. In such a case, the planet is not breathing. It is drying out in a chemically dramatic fashion. That distinction is slightly important. This is why false positives are not minor annoyances. They are the whole reason the field has to be rigorous. A claim of life beyond Earth would be historic, and a claim that large cannot rest on one gas, one spectrum, or one appealing interpretation. Scientists have to treat rival explanations seriously, not as obstacles to their preferred conclusion, but as the actual path toward credibility. A biosignature claim becomes strong only when geological, photochemical, and atmospheric alternatives have been tested hard and have failed to account for the evidence. This work depends on models as much as on measurements. Planetary climate models help researchers understand how atmospheres circulate, warm, cool, and respond to stars. Photochemical models simulate how light and gases interact, what molecules should be produced or destroyed, and what stable states a planet might settle into. Laboratory simulations test how minerals, gases, radiation, and temperature interact under plausible planetary conditions. Comparative planetology brings all of this together by using the worlds of the solar system as reference cases. Mars, Venus, Titan, Europa, Enceladus, Earth, and the giant planets each show different combinations of atmosphere, chemistry, radiation, geology, and environmental history. Research groups linked to NASA, the European Space Agency, and university laboratories rely on this broad toolkit precisely because life detection cannot be solved by observation alone. It has to be interpreted through physically realistic alternatives. Mars is an excellent example of why that matters. Again and again, Mars has offered evidence that sounds tantalizing. Ancient lake beds, organics in rock, methane reports, reactive chemistry and soil, and long-standing debates about experiments from the Viking missions have all made the planet seem biologically tempting at various times. Yet every one of those clues has remained entangled with non-biological explanations. Martian geology, radiation chemistry, perchlorates in the soil, and atmospheric complexity all make the picture harder to read. Mars is not disappointing because it refuses to confirm life quickly. Mars is valuable because it demonstrates how easily a planet can produce partial signals without yielding a final answer. Venus provides another cautionary tale. For a brief period, phosphine in the atmosphere of Venus became one of the most talked about possibilities in planetary science. On Earth, phosphine can be associated with biology in certain environments, so the initial announcement drew enormous interest. But the signal quickly became controversial. Questions arose about the data analysis, calibration, spectral interpretation, atmospheric chemistry, and whether the claimed abundance was real at all. Alternative explanations were explored, and the original excitement weakened under scrutiny. That does not make the episode a failure. It makes it a demonstration of how the process should work. An extraordinary suggestion was met with aggressive testing, and the burden of proof remained where it belonged. The same issue appears in exoplanet atmosphere interpretation. A molecule detected in a distant atmosphere may look significant, but the spectrum may be noisy, clouds may be masking parts of the atmosphere, the host star may be contaminating the signal, and several different combinations of molecules may fit the data almost equally well. This is one reason the field puts such emphasis on retrieval models, repeated observations, and cross-checking with different instruments. The problem is not only measuring the atmosphere, it is deciding whether the measured pattern truly demands biology or merely allows it as one possible interpretation. Comparative planetology helps keep scientists honest here. Venus warns that greenhouse climates and atmospheric chemistry can create extreme worlds without life. Mars warns that habitable conditions and organic compounds are not enough. Titan warns that rich carbon chemistry does not automatically mean biology. Earth warns that life can be real long before it becomes atmospherically obvious. Each world sharpens the broader lesson. No single planetary feature should be treated as a shortcut to conclusion. So when scientists talk about abiotic imposters, they're not being pessimistic. They're doing the work required to make any eventual claim believable. A genuine discovery of extraterrestrial life would need to survive contact with all these alternative explanations. It would need to look convincing not only in one data set, but across time, across models, and ideally across multiple methods of observation. It would need environmental context, chemical context, and consistency. That is why serious researchers do not rush from an unusual signal to a public declaration. They look for multiple layers of evidence. They want repeated measurements. They want different instruments to agree. They want geological and atmospheric models that cannot easily reproduce the same pattern without life. They want the case to strengthen as scrutiny increases, not weaken. Nature is very good at producing suggestive illusions. The real task is not to avoid being excited by them, but to make sure excitement never outruns explanation. Only when a signal survives that level of testing does it begin to move from interesting to credible. And only then would anyone dare treat it as a real sign that a world might be alive. There's a danger in the search for life that receives less attention than false positives, though it may be just as important. A planet can be alive and still look almost dead from far away. It can host biology that is sparse, buried, slow, chemically quiet, or hidden beneath layers of ice, rock, or ocean. In that case, the problem is no longer that nature is faking life. The problem is that real life is being far too discreet. This may be the more humbling possibility, because it suggests that inhabited worlds could exist without announcing themselves clearly to distant observers. Earth itself is the best reason to take that possibility seriously. For much of its history, this planet was inhabited long before it became obvious, in the dramatic atmospheric way modern astrobiology often hopes for. Life emerged early, but the Earth of those ancient ages did not necessarily project a strong, unmistakable signal to a distant telescope. The atmosphere lacked large amounts of oxygen for a vast span of time. Land plants did not yet exist. Complex multicellular ecosystems were still far away. A living Earth might therefore have looked, from sufficient distance, like a chemically modest world rather than a thriving biosphere. That is a sobering thought. If Earth itself could have hidden its biology for so long, other inhabited planets may do the same. This is where the idea of weak biospheres becomes important. Not every inhabited world needs to be lush, productive, and globally transformative. Some may support only microbial life. Some may have ecosystems limited to specific regions. Some may be dominated by low-productivity environments where life survives, persists and evolves, but does not greatly alter the whole planet. A biosphere like that would still be real, still scientifically astonishing, and still very difficult to detect. Life does not need to be abundant to exist. It only needs to endure. Subsurface biospheres are a perfect example. On Earth, microbes live deep below the surface in rock pores, sediments, and fracture networks, cut off from sunlight and often sustained by slow chemical reactions. These organisms are not decorating the planet with obvious colour or flooding the air with spectacular gases. They are simply surviving in the dark using whatever energy gradients are available. If a similar style of life dominated another world, especially one with harsh surface conditions, it might barely affect the atmosphere at all. A distant observer could be staring at a living planet and seeing almost nothing biologically distinctive. The same is true of hydrothermal vent communities in Earth's oceans. These ecosystems do not rely on sunlight. They rely on chemosynthesis, with microbes extracting energy from chemical compounds released where hot water meets rock on the sea floor. Entire communities can exist in darkness, thriving in a setting that would have seemed absurdly hostile by older standards. Yet if ocean-covered worlds create their own version of this problem, a planet with a global ocean may be habitable and even inhabited, but if there are few exposed continents, limited nutrient cycling at the surface, or only weak atmospheric exchange, its biosignatures may remain muted. The biosphere could be active below the surface waters, while doing very little to the atmosphere that astronomers are trying to read. A buried biosphere under ice is even harder. Europa could, in principle, host life in a dark ocean and still give only faint or indirect signs. The same is true for any world where biology is trapped beneath a thick barrier between the organisms and the observable atmosphere. This is why detectability and habitability are not the same thing. A world can be excellent for life and terrible for observers. In fact, one could argue that life itself may often prefer privacy. Surface conditions can be dangerous. Radiation, impacts, drying, freezing, and chemical extremes all make hidden environments attractive. Rock, water, and ice provide shielding. Subsurface habitats offer stability. From the point of view of a microbe, being difficult for astronomers to detect is not a flaw. It is often a sensible lifestyle choice. The possibility of chemically quiet life is especially important. Many biosignature discussions focus on gases because gases are observable from great distance. But not all life produces strong atmospheric consequences. A sparse biosphere may recycle materials locally without building large global imbalances. It may exist in equilibrium with its environment so efficiently that it leaves only subtle traces. In that case, a planet could seem almost inert even though metabolism, reproduction, and evolution are quietly unfolding somewhere beneath the surface or under the sea. That forces a change in strategy. If life may be subtle, hidden, or globally quiet, then astronomers cannot rely only on looking for loud atmospheric markers. They also benefit from watching how worlds change over time. Time-dependent behavior can be revealing. Seasonal shifts in gas levels, repeating patterns in surface reflectance, cyclic changes in clouds or chemistry, and other forms of planetary variability may offer clues that a single snapshot would miss. A still image of a world might look lifeless. A sequence of observations might show that something active is happening. So the problem is not only that nature can imitate life, it is also that life can imitate inactivity. A planet may be inhabited and yet seem geologically dull, atmospherically ordinary, or chemically unremarkable from far away. Earth's early history proves that this is not merely theoretical. The search for life therefore has to remain open to the possibility that many inhabited worlds will look ambiguous, faint, or almost inert until observed with enough patience and over enough time. That makes the next step especially important. If alien life may not advertise itself through obvious gases alone, then one of the best ways to notice it may be through patterns, cycles, and changes that unfold across time, rather than through one dramatic chemical clue seen once. A single observation can tell astronomers what a planet seems to be. A sequence of observations can begin to show what the planet is doing. That distinction matters because life is not simply a collection of chemicals sitting in one place. Life is activity. It takes in energy, transforms matter, responds to changing conditions, and often does so in repeating patterns. If a distant world changes over time in ways that are difficult to explain through simple physics or chemistry alone, those changes may become more revealing than any one still image of its atmosphere. This is why time matters so much in life detection. A static composition can be interesting, but dynamic behaviour is often more persuasive. One measurement might show methane, oxygen, haze, or some unusual surface reflectance. That is useful, but limited. It is like walking into a room, seeing a table laid out for dinner, and trying to reconstruct the entire evening from one glance. If the same room is observed again and again, patterns begin to emerge. The lights dim at certain times, steam rises, plates move. Someone is clearly doing something. A planet can be like that. Repeated observations can reveal that its chemistry and surface are not simply existing. They are behaving. Seasonal swings are one of the clearest examples. On Earth, biology contributes to large seasonal changes in atmospheric carbon dioxide, especially across the northern hemisphere, where plant growth rises and falls with the changing seasons. Surface reflectance changes as vegetation grows, dries, expands, or retreats. Biological productivity is not constant throughout the year, and the atmosphere records some of that rhythm. If a distant planet showed repeating chemical changes tied to its orbit around its star, astronomers would pay very close attention. A world whose gases rise and fall in a regular seasonal pattern is telling a more active story than a world described by one averaged spectrum. That does not mean seasonal behaviour would automatically prove life. Non-biological processes can be seasonal too. Ice can sublime and refreeze. Clouds can shift with temperature. Photochemical reactions can change with illumination. Surface materials can appear darker or brighter as frost moves or dust is redistributed. But even so, time-dependent behaviour is valuable because it adds structure to the evidence. It allows scientists to ask not just what is present, but how it changes, when it changes, and whether those changes fit a coherent physical explanation. Methane is one gas that often appears in these discussions because it is chemically interesting and potentially variable. Seasonal methane variation, if robustly detected on another world, would immediately raise questions. Is the methane being produced steadily and then redistributed by the atmosphere? Is it being released from subsurface reservoirs when temperatures change? Is chemistry in the atmosphere destroying it at a variable rate? Or could biology be contributing to the cycle? The value of the pattern lies not in offering an instant answer, but in narrowing the possibilities and forcing models to explain not only the gas itself, but its changing behaviour. Surface reflectance changes matter for similar reasons. Different surfaces reflect light differently, and life can influence that reflection. On Earth, large-scale vegetation creates patterns that shift with season and geography. A distant world might show broad changes in brightness, or colour linked to surface chemistry, frost, biological pigments, or something else entirely. The signal would be subtle, certainly, and difficult to interpret, but it would add another layer to the puzzle. A changing surface is more informative than a fixed one because change reveals process. This is why long-term monitoring becomes so important. One observation can be noisy. Two observations can disagree for reasons that have nothing to do with the planet. But repeated campaigns over many orbits allow astronomers to build confidence. They can begin to separate signal from noise, one-off anomalies from genuine cycles, and instrument quirks from real planetary behaviour. Long-term monitoring is not glamorous, which may be why it rarely gets invited to the dramatic public paintings of alien worlds. But it is one of the strongest ways to turn speculation into a real scientific case. Future observatories will matter enormously here. Some of the most interesting planetary behaviour may only become visible when worlds are watched repeatedly, with enough precision and over long enough timescales. That requires patience, telescope time, and instruments capable of detecting weak variability rather than only strong one-time signals. For many exoplanets, astronomers may need to observe transit after transit, eclipse after eclipse, and seasonal cycle after seasonal cycle before they can say with confidence that the atmosphere truly behaves in a patterned way. Statistical confidence becomes central in that kind of work. Weak signals are dangerous because they invite wishful thinking. A tiny change in a spectrum may be real, or it may be instrument noise, stellar contamination, data reduction error, or ordinary bad luck. So scientists need enough repeated evidence to show that the pattern is not a coincidence. The signal must rise above noise not once, but reliably. It must appear where expected, disappear when expected, and survive different methods of analysis. A persuasive pattern is not merely observed, it is replicated through time. This brings the whole search back to one of its deeper principles. Life is not just matter, it is process. A living world may reveal itself not through one spectacular atmospheric ingredient, but through a stable pattern of change that ordinary chemistry struggles to imitate. A gas that rises and falls with regularity, a surface that shifts in a repeating way, an atmosphere that never settles into simple equilibrium. All of these become more meaningful when they persist over time. The case strengthens not because one clue becomes louder, but because several clues begin behaving like parts of one active system. Even then, caution remains necessary. A suggestive world seen from afar is still only suggestive. The scientific community would need clear standards for deciding when repeated patterns, chemical signals, and environmental context add up to something worthy of public announcement. That threshold cannot rest on excitement alone. It has to depend on evidence becoming strong enough, stable enough, and resistant enough to alternative explanations that the claim can survive serious scrutiny. So time does not merely add detail to life detection, it changes the whole quality of the evidence. A still planet may be interesting, a changing planet may be telling a story. And once that story begins to look coherent, repeated, and difficult to explain away, the question is no longer simply what the world contains. It becomes whether the world is doing something so organized and persistent that science has to start taking the possibility of life very seriously indeed. At this point, the question becomes less romantic and more practical. What would scientists actually accept as real evidence of life? Not exciting evidence, not suggestive evidence, not the kind of evidence that produces a dramatic headline on a Tuesday and a careful correction by Friday. Real evidence has to survive skepticism. It has to hold together when people who are highly trained, deeply cautious, and professionally suspicious start trying to break it apart. That means one thing immediately. A single clue would almost never be enough. One gas in an atmosphere would not do it. One odd rock texture would not do it. One puzzling seasonal pattern would not do it. Real confidence would require multiple biosignatures pointing in the same direction, gathered in a setting where the environment makes biological interpretation plausible, and where major abiotic explanations have been tested hard and found lacking. In other words, scientists would want convergence. Different kinds of evidence measured in different ways, all leaning toward the same conclusion. Environmental context matters because no signal means anything on its own. Oxygen is interesting, but oxygen around a planet losing water to space might be misleading. Methane is interesting, but methane produced by water-rock reactions is not life. Organic molecules are interesting, but chemistry can make organics without help from biology. So before anyone serious would start speaking about life, they would want to know the temperature, the host star, the likely atmospheric structure, the surface or subsurface environment, the geological activity, and the broader chemical setting. A signal only becomes stronger when it fits into a world where biology makes more sense than the alternatives. This is why scientific language becomes so careful. A planet may be called potentially habitable if its size, orbit, and temperature range suggest that liquid water might be possible. That does not mean life has been detected. It does not even mean the atmosphere is known. It simply means the world belongs on the list of places worth paying attention to. A biosignature candidate is more specific. It means some measured feature could be related to life, but the evidence remains preliminary or incomplete. Evidence consistent with life is stronger still, but even that phrase is cautious. It means the observations fit a biological explanation without claiming that biology has definitely been shown. Confirmed detection is a much higher standard. That would imply the evidence has survived repeated scrutiny, independent analysis, alternative explanations, and further observation, and that process would be social as well as scientific. Science is not one person looking through a telescope, gasping softly, and immediately being believed by the planet. It is a collective process. Data would be checked by the instrument team, analysed by multiple researchers, and tested against different models. Other groups would attempt their own retrievals and their own interpretations. Papers would go through peer review. Critics would question the assumptions, the calibration, the statistics, the atmospheric models, and the handling of noise. Major observatory teams would be cautious. Institutions such as NASA, the European Space Agency, and large university collaborations would almost certainly avoid overstatement until the case became unusually strong. That caution is not a flaw. It is the reason any eventual claim would matter. Responsible scientists are reluctant to overstate ambiguous findings, because the history of science is full of things that looked extraordinary for a moment, and then became ordinary, mistaken, or at least much less exciting after closer inspection. Mars taught that lesson. Venus taught it too. Exoplanet atmosphere studies keep teaching it. Extraordinary claims do not fail because science is timid. They fail because planets are complicated, data are noisy, and nature is extremely creative when it comes to producing confusion. So a single dramatic headline would almost certainly not settle the question. More likely, the first serious sign of alien life would arrive as a gradual shift in scientific confidence. At first there might be a strong paper suggesting an unusual combination of gases or a pattern difficult to explain abiotically. Then, follow-up observations would come in. Other teams would test the result. Some would disagree, some would propose alternative explanations. More data would accumulate. Perhaps a second instrument would see the same thing. Perhaps long-term monitoring would reveal a repeating pattern that strengthens the biological interpretation. Confidence would build by degrees, not by thunderclap. This slow accumulation is exactly what real evidence looks like in difficult sciences. It is not cinematic, but it is trustworthy. The strongest case would probably include several features at once. Multiple biosignatures, a chemically plausible setting for life, a star and atmosphere that do not make easy abiotic explanations more likely, reproducibility across observations, agreement across instruments, where possible. Continued survival of the interpretation after competing models try to replace it. That is the kind of case that would start to feel real. Even then, language would remain careful for a long time. Scientists might say that a planet shows the strongest evidence yet for biological activity. They might say the observations are best explained by a living process, while still leaving room for some unknown alternative. Full certainty might arrive only very slowly, and in some cases it might remain slightly out of reach for years. That can sound frustrating, but it is also honest. The universe does not owe anyone a neat answer. What this means in practice is that a discovery of life beyond Earth would probably not look like a clean instant revelation. It would look like a growing imbalance between explanation and doubt. The biological interpretation would keep surviving. The non-biological alternatives would keep weakening. Over time, the balance would shift until the scientific community began to treat life as the most credible explanation rather than merely one exciting possibility among several. And once that happens, the question changes. It is no longer only about method, spectra, gases, or models. It becomes about meaning. What would it feel like in real human terms to realize that somewhere beyond Earth, another world is not just habitable, not just interesting, but genuinely alive? If the signal came tomorrow, it would probably not arrive in the form people have been trained to expect by fiction. There would be no clear photograph of a creature standing on a ridge and looking mildly annoyed at the camera. There would be no radio message saying hello in excellent grammar. The most realistic near-future scenario would be much quieter and much stranger. It would likely be an atmospheric pattern in the light from a distant planet, a set of chemicals in a plume from an icy moon, or a rock sample containing textures, isotopic ratios, and organic compounds that together begin to resist every non-biological explanation. That is important to say plainly, because the first evidence of life beyond Earth is overwhelmingly more likely to suggest microbes than anything dramatic. Microbial life is simpler, older, and more chemically plausible across a wide range of environments. On Earth, microbes dominated the story of life for most of the planet's history. Complex multicellular organisms came much later, and intelligent life later still. So if a real signal arrives soon, the odds strongly favour something indirect and microbial. Perhaps a pattern of gases in an exoplanet atmosphere that looks chemically maintained in a way geology struggles to reproduce. Perhaps material from Enceladus showing a combination of organics, isotopic ratios, and disequilibrium chemistry that starts looking less like a hydrothermal system alone and more like a living one. Perhaps a Martian sample that, under repeated examination, shows a network of clues too coherent to dismiss. If that happened, the scientific response would not be immediate certainty. It would be analysis, then reanalysis, then fresh attempts to prove the first interpretation wrong. The team that made the initial result would check the instrument, the calibration, the statistics, and the retrieval models. Other researchers would go through the data independently. Rival groups would test different explanations. Telescope time would suddenly become even more valuable than usual, which is saying something, because astronomers already treat good telescope time the way dragons treat treasure. Follow-up observations would be scheduled if possible. Laboratory modelling would expand. Planetary chemists, geologists, atmospheric scientists, microbiologists, and instrument specialists would all begin circling the problem from different directions. If the signal came from a plume or a returned sample, laboratories across the world would begin a careful sequence of checks. Could contamination explain it? Could mineral processes mimic it? Could isotope fractionation occur abiotically under those conditions? Could the textures have formed through non-biological chemistry? If the signal came from a distant atmosphere, observatories would try to measure it again. Other instruments would be asked to confirm the same gases or look for related ones. Models would be rerun under different assumptions. Stellar contamination would be examined. Photochemistry, volcanic activity, atmospheric escape, and every other abiotic possibility would be forced to take their turn. Public communication would also be cautious, or at least it would try to be. That part is not trivial. Scientific institutions such as NASA, the European Space Agency, major observatory teams and university collaborations would know immediately that a claim of extraterrestrial life is unlike almost any other announcement. Every phrase would matter. Words like detected, evidence, candidate, consistent with, and confirmed would be weighed very carefully. Some people would inevitably run ahead of the data. Others would be so cautious they would seem almost unreasonably reluctant. That tension would be normal. It would not mean the evidence was weak. It would mean the stakes were immense, and the stakes really would be immense even if the life in question turned out to be microbial. In public imagination, alien life often means intelligence, technology, communication, or something large enough to require impressive music in the background. But scientifically, microbial life would already be revolutionary. It would mean that biology is not unique to Earth. It would mean that the transition from chemistry to living systems happened at least twice in the universe. That alone would transform biology, because it would tell us that life is not a singular accident known only from one world. It would reshape planetary science because planets and moons would no longer be studied only as places where life might be possible, but as places where life has actually happened. It would also change the philosophical picture in a very quiet but profound way. Earth would stop being the sole known location where matter became aware enough to ask questions about itself. That does not require little green visitors or galactic diplomacy. It does not even require complex cells. A confirmed extraterrestrial microbe, or a confirmed atmospheric biosignature pointing strongly to microbial activity, would be enough. In some ways, that result would be even more powerful, because it would be so basic. It would say that the universe can produce life without needing to produce anything dramatic. Biology would move from being a local fact to a cosmic process. There would also be a slower emotional side to it. At first, there would be excitement, confusion, arguments, overinterpretation, and some very confident television discussions by people who had not read the paper properly. But after the noise settled, something deeper would remain. A second origin of life would make the universe feel at once more populated and more continuous. The distance between Earth and the rest of nature would shrink a little. Life would no longer seem like a lone flare in a dark and indifferent cosmos. It would begin to look like one expression of something the universe can, under the right conditions, do more than once. Still, even if the signal came tomorrow, scientists would not claim to know lightly. They would know in the way science comes to know difficult things, through converging evidence, through repeated tests, through disciplined doubt, through the gradual elimination of alternatives, through the refusal to confuse excitement with certainty. That is the real answer to the title. There is no single shortcut, no one perfect gas, no one spectacular image that would instantly settle the matter. There is only the patient effort to tell life apart from the clever tricks of chemistry, and to keep going until the biological explanation is the one that remains standing. So if the signal came tomorrow, the beginning would probably be subtle, a pattern in light, a strange chemical imbalance, a sample that looks just a little too organized to be innocent. And then, slowly, after scrutiny, argument, modeling, repeat observations, and the hard work of ruling out every easier answer, the possibility would start to harden into knowledge. Not simple knowledge, not effortless knowledge, but real knowledge all the same. So, how would scientists actually know if there is life beyond Earth? Not through one magical sign, not through one dramatic headline, and almost certainly not through a single photograph, one suspicious gas, or a lonely rock that looks just a bit too interesting for its own good. They would know by building a case slowly, carefully, and piece by piece. Biosignatures would matter, but only in context. A gas in an atmosphere would need to make sense alongside the star, the climate, the geology, and the rest of the chemistry. A plume from an icy moon would need to show more than organics alone. A sample from Mars would need to survive every non-biological explanation that chemistry, physics, and geology could throw at it. That is why Earth has remained so important throughout this whole question. It is the only confirmed example of a living world, and it shows both what life can do and how difficult life can be to recognize from afar. Mars teaches caution. Ocean worlds such as Europa and Enceladus widen the idea of habitability. Titan warns against assuming that life must look like Earth's version. Exoplanets push the challenge even further, because their atmospheres must be read from faint changes in light alone. Across all of this, one principle keeps returning. The strongest evidence would not be one isolated clue, but a pattern of chemical disequilibrium, environmental plausibility, repeat observations, and signals that remain convincing after every abiotic alternative has been tested. In that sense, the search for life is really a search for reliable evidence of active biology. It demands patience because nature is good at imitation. Volcanoes, radiation, water-rock reactions, atmospheric escape, and photochemistry can all produce fragments of the story without life ever appearing. At the same time, real life may be quiet, sparse, buried, or hidden beneath oceans, ice, or rock. A world may be inhabited and still look almost silent from a distance. So the universe may be full of habitable places, perhaps even full of living ones, and the hardest part may not be that life is rare. The hardest part may be learning to recognize it honestly. No wishful thinking, no rushing ahead, no mistaking chemistry for biology, and no overlooking biology because it first appears as something faint and ambiguous. One of the greatest discoveries in human history may not arrive as a shout. It may arrive as a subtle pattern in light, chemistry, ice, or stone that survives every attempt to explain it away. And when that happens, that is how scientists will know.